हेलो दोस्तों, कैसे हैं आप लोग। उम्मीद करते हैं आपके एग्जाम्स अच्छे हो रहे होंगे। जैसा हमने प्रॉमिस किया था आपको, आज डिस्कस करेंगे हम मोस्ट एक्सपेक्टेड पूल ऑफ बायोकेमिस्ट्री क्वेश्चंस। हमने इन क्वेश्चंस को सेक्शंस में डिवाइड किया है, जिससे कि यू कैन इजीली कॉन्सेंट्रेट ऑन द इंपॉर्टेंट टॉपिक्स। तो शुरू करते हैं। सबसे पहला जो टॉपिक है वो क्वेश्चंस इन्वॉल्व करेंगे बायो कैलकुलेशन से। ठीक है, तो शुरू करिए। Molarity of a solution is the number of, that's very easy. A molarity is a concentration unit which defines as a number of moles of a solute in a liter of a solution. So option B is the correct answer. The density of a solution prepared by dissolving 120 gram of urea, जिसका molecular mass दे रखा है 60, in 1000 gram of water is 1.15 gram per ml. The molarity of this solution is, तो इसमें बड़ा सिंपल सा है, पहले आपको सॉल्यूट की मोलैरिटी निकालनी होगी, फिर सॉल्यूशन की निकालिए, और उसके बाद अगर आप कैलकुलेट करते हैं तो जो मोलैरिटी है एज पर द सॉल्यूशन, इसका आंसर आएगा 2.05, दैट इज ऑप्शन बी। द वैल्यू ऑफ आयनिक प्रोडक्ट ऑफ वाटर एट 25 डिग्री सेल्सियस इज, ऑलवेज रिमेंबर द आयनिक प्रोडक्ट ऑफ वाटर is heavily dependent on the temperature. As you can see it in the table, the value of ionic product changes as the temperature is increased, and hence at 25 degrees Celsius this value comes around as 10 to the power minus 14, which is close to option C. pH of an aqueous solution of a 1 molar NaCl would be acidic, neutral, alkaline, or cannot be determined. When a strong acid and a strong alkali they combine, they form neutral salts. For example, whenever HCl combined with the NaOH, it forms NaCl and H2O. Both do not affect the pH, hence the pH of the water would be 7, that is neutral. So option B would be the correct answer, that whenever we dissolve 1 molar NaCl in water or any solution, it won't change the pH and the pH of the solution will remain. That means pH of the water will be neutral. Determine the pH of a 0.01 normal NaOH solution. इसको तो बहुत आराम से आप सॉल्यूशन को निकाल सकते हैं कि pH ऑफ 0.01 नॉर्मल जो NaOH या फिर 10⁻⁻² मोलर NaOH जो होगा उसको हम कैलकुलेट करेंगे। पहले आप जो NaOH दिया हुआ है तो उससे आप POH निकाल लीजिए, और वंस द यू गॉट द POH तो आप उसे 14 में से माइनस कर दीजिए तो आपका pH निकल आएगा। तो इसका ऑप्शन होगा ऑप्शन डी, दैट इज द pH ऑफ 0.01 नॉर्मल NaOH वुड बी 12. HCl solution of a 1 millimolar was diluted to 10 to the power 6 times. What would be the pH of a resulting solution? अगर किसी HCl solution को अगर हम 10 to the power 6 times अगर डाइल्यूट कर देंगे तो उसमें आपको ध्यान रखना है कि इसमें हमें वाटर का कंट्रीब्यूशन भी लेना पड़ेगा प्रोटॉन्स के लिए, क्योंकि वाटर के भी अपने प्रोटॉन्स होते हैं, और अगर जब हम टोटल प्रोटॉन्स निकालेंगे तो उससे pH जो निकल के आएगा which will be around 7. तो ऑप्शन बी should be the correct answer for this question, that pH 7 would be the right pH for this solution. The approximate molecular weight in kDa of the product after the translation of a 390 basis mRNA will be 48 kd, 26 kd, 39 kd, or 40 kd. As we know that the number of base pair is given as 390 and each codon is having 3 base pair. That means total codon would be around 130. 390 divided by 3. Now, an individual amino acid has a molecular weight of 110 dalton. So, a peptide of having 130 amino acid will give me 14300 dalton or 14.3 kilodalton protein. So, option D has to be the correct answer. बायोमोलिक्यूल्स कैटेगरी में अमाइनो एसिड्स जो हैं वो बहुत इंपॉर्टेंट टॉपिक्स है और बहुत सारे क्वेश्चंस आए हुए हैं पास्ट ईयर में। तो हम आपको रिकमेंड करेंगे कि अमाइनो एसिड्स की टेबल और उसके इंपॉर्टेंट वन लाइनर्स को जरूर रिफ्रेश करके जाइएगा। ओके, द बॉन्ड एंगल बिटवीन HOH इन वाटर मॉलिक्यूल इज 55 डिग्री, 105.4 डिग्री, 104.5 डिग्री और 110 डिग्री. If you can see in the diagram, the bond angle in water is 104.5 degree due to the presence of two lone pair of electrons. So option C is the correct answer. Which of the following bonds inside a cell can be formed by the COH group of cellular amino acids? amide and carboxylic anhydride bonds, ether and ester bonds, amide and ether bonds, or ester and amide bonds. Each amino acid has two functionality, amino group and COH group. There are certain reactions which are due to the COH group, that is like salt formation with alkalis, ester formation with alcohols, amide formation with amino group, and decarboxylation. While the reaction specific for NH2 groups are deamination reaction and reaction with ninhydrine chemical. So here the option D has to be the correct answer, that the reaction specific to COH group present in any amino acid would be ester formation with alcohols and amide formation with NH2 groups. Formation of a disulfide bond between two cysteine residues requires which of the following? Oxidizing agent, adenosine triphosphate, reducing agent, or ammonium sulfate. Generally, reducing agent like beta-mercaptolithanol or dithioethol may facilitate the formation of disulfide bond between two cysteine residues. So option C is the correct answer. Which of the following statement is true? RNAs can be treated with urea to produce a random coil. If one oxidizes random coil RNAs in urea, it quickly regains its enzymatic activity. If one removes the urea and oxidizes RNA slowly, it will renature and regain its enzyme activity. Although renatured RNAs has enzyme activity, it can be readily distinguished from naive or native RNAs. Basically, this question is talking about the Anfinsen experiment. What he did? Actually, he took a native RNA and given the treatment of urea and beta-mercaptolithanol. Once this treatment is being given, definitely RNase has lost all its activity. Once they dialyze the urea and beta-mercaptolithanol, that means they have removed urea and actually removed beta-mercaptolithanol, we found that the RNase actually regain its all activity, almost more than 95% of its activity. So basically, what he concluded that a denatured form actually gets folded because of the presence of its amino acid sequence, which was the landmark statement as far as protein folding is concerned. So basically in this, the option C is the correct answer, that if one removes the urea and oxidizes RNase, that means remove the beta-mercaptolithanol, then RNase will renature and regain its enzymatic activity. Which group of a fully protonated glycine will release the proton first when titrated against hydroxyl ion? Amino group, cannot be predicted, both at the same time, or carboxylic group. Generally, at an alkaline condition, the carboxylic group, they release their proton easily. And once all the protons are being extracted from a carboxylic group, if at all, then generally, as a rule, amino group gets deprotonated. So generally carboxylic group in any amino acid, they release their proton first and followed by the amino group. So option D is the correct answer. All the below given amino acids are optically active except glycine, phenylalanine, valine, and tryptophan. As we have just discussed that glycine do not have any asymmetric carbon, hence it has to be optically inactive. So option A has to be the correct. Select the amino acid which can exist as a diastereomer: valine, proline, glycine, or isoleucine. Stereoisomers that are non-superimposable, non-mirror image are known as diastereomers, and generally this phenomena is observed when a chemical have more than one chiral center. Isoleucine and threonine has got two chiral centers, while glycine has none. So isoleucine and threonine will be existing as more than one diastereomers, while glycine will never produce a optically active compound. The following amino acid is least likely to be found in a alpha helix structure. बड़ा सिंपल सा क्वेश्चन है और याद रखिए ये क्वेश्चन आपको कई एग्जाम्स में मिलेगा. प्रोलिन जो होता है वो एक ऐसा अमाइनो एसिड है जो अल्फा हेलिक्स के इन बिटवीन कभी नहीं मिलेगा. इनफैक्ट प्रोलिन को हेलिक्स ब्रेकर भी बोला जाता है. तो यानी प्रोलिन की जो प्रोपेंसिटी है मिलने की अल्फा हेलिक्स में वो मिनिमम है या फिर होगा ही नहीं. ग्लाइसिन इज अनदर अमीनो एसिड जो कि लीस्ट लाइकली है किसी भी अल्फा हेलिक्स में मिलने के लिए। तो इसमें ऑप्शन जो होगा वो होगा ऑप्शन डी। आगे चलते हैं। Which of the following is an essential amino acid for human? Serine, alanine, isoleucine, or aspartic acid? Remember, there are 9 amino acids like histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. We are repeating again. Histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. These are the essential amino acids and therefore indispensable element and absolutely required through our diet. So option C is the correct answer, that isoleucine is an example of essential amino acid for humans. Which amino acid of a protein can make shift base with a reducing sugar? Glycine, lysine, tryptophan, or phenylalanine? Generally, any amino acid with a free amino group like lysine may react with a reducing sugar to form the shift linkage. So option B is the correct answer. The biosynthesis of proline takes place from which amino acid? तो एक्चुअली अगर हम ध्यान से देखें तो ग्लूटामेट से प्रोलिन की सिंथेसिस स्टार्ट होती है। ग्लूटामेट इज फर्स्ट कन्वर्टेड इनटू पायरोलिन कार्बोक्सिलिक एसिड, जिसे हम P5C भी बोलते हैं। और ये जो एंजाइम, ये जो रिएक्शन है, ये कैटलाइज करी जाती है दो एंजाइम के थ्रू। P5CS जो है वो ग्लूटामेट को कन्वर्ट करता है पायरोलिन 5 कार्बोक्सिलिक एसिड में, which is again being converted into proline by P5CR in the chloroplast or cytosol. So hence the answer would be glutamate acid. Amino acid residues which are predominantly involved in the protein DNA interactions are alanine, negatively charged, prolines, or positively charged. See, DNA is negatively charged due to the phosphate backbone. So proteins interacting with the DNA must have positively charged or basic amino acid like lysine, arginine, or histidine. So the amino acids has to be the positively charged as present in even histones. So D has to be the correct answer. Which of the following is a polar amino acid? Threonine, is it alanine, proline, or methionine? Basically, polar amino acids are those amino acids which are having a side chain that prefer to reside in aqueous environment and generally exposed over the protein surfaces. 6 amino acids which are polar uncharged, that means they do not carry the charge, are serine, threonine, cysteine, asparagine, glutamine, and tyrosine. So out of the given option, threonine is the correct answer. Natural proteins are composed of 20 alpha amino acids. Which one of the following statements is true for any of these amino acids in a solution of pH 1.5? Only amino group is ionized, only carboxylic group is ionized, both amino and carboxylic groups are ionized, or both the amino and carboxylic groups are neutral. Always remember, every amino acid generally develops a net positive charge when it is kept at acidic pH, where amino group is protonated, and net negative charge at high pH because the carboxylic groups gets deprotonated. So here the option A has to be the correct answer, that whenever any amino acid is kept at a very low pH, then only amino group is ionized. All the following statements about phenylketonuria are correct except. So basically what happens, the person who are suffering from phenylketonuria actually has a deficiency of phenylalanine hydroxylase. This enzyme is basically required for the synthesis of tyrosine from phenylalanine. So here the option A is correct, that phenylalanine cannot be converted into tyrosine in the case of phenylketonuria. Now, since phenylalanine is found in all protein-rich foods, hence the person who is suffering from phenylketonuria must permanently follow a low protein or phenylalanine diet. That means option C is also correct. Now, the patient can be diagnosed for the disease by measuring urinary metabolites like phenylalanine, phenyllactate, phenylacetate, phenylpyruvate, or even phenylethylamine. So hence option B is also correct. So that means in the case of this question, Option D is the correct answer. That this statement, option D, it leads to the decreased synthesis of thyroid hormone, catecholamine and melanin, is not a characteristic of phenylketonuria. Maple syrup urine disease is due to the metabolic defects in the pathways of degradation of MSUD जो है या मेपल सिरप यूरिन डिजीज जो है वो एक इनबॉर्न एरर ऑफ मेटाबॉलिज्म है प्रोटीन का which is happening due to the deficiency of branch chain alpha keto acid dehydrogenase complex which is an enzyme. और जब ये होती है डिजीज तो इट लेड टू द बिल्डअप ऑफ ब्रांच चेन अमाइनो एसिड विच इज ल्यूसिन, आइसोल्यूसिन, वेलाइन और उसके बाय प्रोडक्ट्स को. तो इसीलिए इसमें ऑप्शंस होगा ऑप्शन डी, ब्रांच चेन अमाइनो एसिड्स. आगे चलते हैं. The most of the UV absorption of proteins above 240 nanometer is due to their content of. That's very simple. Commonly, the optical absorption of protein is measured at 280 nanometer. At this wavelength, the absorption of protein is mainly due to the aromatic amino acid like tryptophan, tyrosine, and to some extent cysteine. So here in the given question, the option A is the correct answer. A Ramachandran plot represents the sterically allowed conformation of a polypeptide backbone, gives the frequency of occurrence of amino acid in alpha helical and beta sheet structure, predicts alpha helical and beta sheet conformation of a peptides and proteins, or shows the X-ray diffraction pattern of a protein. Ramachandran plot is a 2D plot between the torsional angle phi and psi across the peptide backbone. Basically, this plot is used to visualize sterically hindered or energetically allowed region of a polypeptide. So, a Ramachandran plot basically represents the sterically allowed conformation of a polypeptide backbone. The cooperativity phenomena of protein ligand interaction was first determined in hemoglobin oxygen, peroxidase water, ATPase aspartate, or myoglobin oxygen. See, myoglobin cannot be the answer because myoglobin is a classical example of Michaelis-Menten protein. Hemoglobin, on the other hand, is a classical example of allostery and positive cooperativity. When first oxygen binds to hemoglobin, it has lower affinity, while subsequent binding of oxygen observe higher and increased affinity. Hence, hemoglobin is a model protein to understand allosteric behavior of any protein. Which one of the following is not a heme protein? Catalase, tryptophan pyrrolase, delta aminolevulinic acid synthetase, or cytochrome oxidase. Heme containing proteins, they contain a special chemical called heme group which confer them with special functionalities like oxygen carrying capacity, oxygen reduction, or even electron transfer capacities. Heme is synthesized in bone marrow or liver with the help of delta aminolevulinic acid synthetase. Hence, the third enzyme actually synthesize heme rather than contain heme. So, option C is the correct answer. Heme containing proteins that function as an electron carrier in oxidative phosphorylation and photosynthesis are hemoglobin, catalases, chlorophyll, or cytochromes. Cytochromes are the heme proteins that are involved in electron transport chain, that is oxidative phosphorylation and photosynthesis. So option D is the correct answer. Which of the following motif is present in calcium binding proteins? Ig superfold, EF hands, solenoids, or zinc finger? As you can see in the diagram, EF hand is a helix loop helix structural domain found in the large family of calcium binding proteins. So option B is the correct answer. Collagen is rich in glutamic acid and glycine, glycine and glutamine, glycine and proline, glycine and alanine. Collagen अपना बहुत फेमस प्रोटीन है बिकॉज ऑफ इट्स ट्रिपल हेलिक्स स्ट्रक्चर, जिसके अंदर 3 लेफ्ट हैंडेड हेलिक्स जो हैं वो एक दूसरे पे रैप करते हैं इन राइट हैंडेड फैशन और विच आर रिच इन ग्लाइसिन एंड प्रोलिन. बेसिकली स्ट्रक्चरल यूनिट जो होती है अमाइनो एसिड्स की वो होती है ग्लाइसिन, प्रोलिन एंड हाइड्रोक्सी प्रोलिन इन अ कोलेजन रिपीट्स. तो इसीलिए कोलेजन जो होगा वो ग्लाइसिन एंड प्रोलिन में बहुत ज्यादा रिच होगा. तो ऑप्शन सी हैज टू बी द करेक्ट. 4-hydroxyproline is abundantly present in keratin, collagen, myoglobin, or hemoglobin. Prolyl 4-hydroxylation, it is the most abundant post-translational modification of collagen. 4-hydroxylation at third proline residue, it increases the stability of the triple helix of the collagen. So option B is the correct answer. Folding of a protein is primarily governed by ionic strength of a solution, presence of branch chain amino acid, primary structure of a protein, or presence of hydrophobic amino acids. Since proteins are stabilized by thousands of non-covalent interactions between amino acids, इसीलिए प्राइमरी स्ट्रक्चर जो प्रोटीन्स का होगा वो बड़ा इंपॉर्टेंट रोल प्ले करेगा क्योंकि वो एक स्पेसिफिक अरे ऑफ अमाइनो एसिड सीक्वेंस होता है और इसीलिए कौन सा अमाइनो एसिड कहां है उसकी पोजीशन बहुत डिटरमाइन करेगी प्राइमरी स्ट्रक्चर में कि क्या वो फैसिलिटेट करेगा सेकेंडरी स्ट्रक्चर्स बनने के लिए या नॉन कोवेलेंट इंटरेक्शन को फैसिलिटेट कर पाएगा और इसीलिए हमेशा याद रखिएगा कि प्राइमरी स्ट्रक्चर ऑफ अ प्रोटीन ड्राइव प्रोटीन फोल्डिंग इसीलिए ऑप्शन जो सी है दैट हैज टू बी करेक्ट आंसर. The chemical nature of covalent linkage in sucrose is known as N-glycoside, O-glycoside bond, amide, or diester. Sucrose is composed of alpha-D-glucose condensed with beta-D-fructose connected with alpha-1,2 linkage, which is a O-glycosidic type bond. So option B is the correct answer. Sucrose is composed of mannose and glucose, galactose and mannose, fructose and galactose, or fructose and glucose. Sucrose is a disaccharide which is made up of glucose and fructose when they are connected through their respective anomeric hydroxyl groups. Due to this reason only, it is a non-reducing sugar. So here the option D would be the correct answer. Which of the following is a non-reducing sugar? Trehalose, cellobiose, maltose, or lactose? If you can see in the figure, the trehalose is a non-reducing disaccharide because here the two D-glucose are linked through their anomeric position to one another. Remember, any monosaccharide which does not have the anomeric hydroxyl group, they would never be a reducing sugar. So here the option A is the correct answer. The trehalose is an example of a non-reducing sugar. Which sugar will give a positive result with Salvinoff test? Mannose, glucose, galactose, or sucrose? Salvinoff test, it is used to distinguish a aldose sugar from ketose sugar. Ketose sugar, they rapidly dehydrated in comparison to aldoses, and upon degradation they react with resorcinol at acidic condition and give a dark red color, while aldoses they give the light pink color. Sucrose is, being a ketose sugar, it is bound to give the positive result with Salvinoff test. So option D is the correct answer. D-glucose and D-mannose are they are the anomers, epimers, ऑप्टिकल आइसोमर्स और डायस्टिरियोमर्स. अगर हम ध्यान से देखें दोनों स्ट्रक्चर्स को, ग्लूकोज का और मैनोज का, ध्यान से देखिएगा तो आप देख सकते हैं कि ग्लूकोज और मैनोज जो हैं वो C2 पोजीशंस पे डिफरेंट है. फिशर प्रोजेक्शंस में अगर कोई भी दो स्ट्रक्चर्स कार्बोहाइड्रेट के स्पेशली हैंडेडनेस ऑफ OH यानी कि एक OH का लेफ्ट हैंड पे है या राइट हैंड पे है, जस्ट इसकी वजह से डिफरेंट होते हैं किसी कार्बन सेंटर पे, तो दे आर ऑफन टर्म्ड एज एपीमर्स. एनोमर्स जो कॉन्सेप्ट है वो हेवर्थ प्रोजेक्शन का सिग्नेचर है. यहां पर जो ऑप्शन बी है दैट हैज टू बी करेक्ट. इसी में एक और क्वेश्चन बनता है ग्लूकोज और गैलेक्टोज के लिए आपको पूछा जा सकता है कभी भी क्वेश्चन, तो जो कि ग्लूकोज और गैलेक्टोज अगर आप ध्यान से देखें तो वो C4 पे डिफरेंट है. तो इसीलिए ग्लूकोज और मैनोज C2 एपीमर्स हैं जबकि ग्लूकोज और गैलेक्टोज C4 एपीमर्स हैं. तो इसीलिए इसका ऑप्शन होगा ऑप्शन बी. As we can see from the past experience, there are a lot of questions which are being repeated from the DU biochemistry exam. So here we are giving the link, you can see it on the top, or we will give you at the last of this video where you can find and check the old videos of our Vigyan Ghar. Now let's begin. The vitreous humor of the eye is composed of chondroitin sulfate, keratin sulfate, hyaluronic acid, or heparin. The vitreous humor is a clear gelatinous substance that occupies the space between the lens and the retina. The natural hydrogel is composed of 98 to 99% of water by weight, and rest is mix of a collagen, hyaluronate, protein, salts and different other sugars. So option C is the correct answer. Which of the following is a ketotriose? That's very simple. Dihydroxyacetone is a natural ketose. Other than that, there is another example of keto sugar is sucrose, but that is an hexose. So option C here is the example of ketotriose. Remember, the aldose version of it is known as glyceraldehyde 3-phosphate. Individuals can have relatively high levels of pyruvate in their blood, which is due to the vitamin B deficiency, vitamin D deficiency, thiamine deficiency और अल्कोहल इंटेक. बड़ा इंटरेस्टिंग सा क्वेश्चन है. अगर आप ध्यान से देखें, जो पायरुवेट है वो कन्वर्ट होता है एसिटाइल कोए में थ्रू एन एंजाइम पायरुवेट डिहाइड्रोजनेज, जिसको के जो यूज करता है कोफैक्टर थायमिन. अगर आपका पायरुवेट डिहाइड्रोजनेज वर्क नहीं करेगा तो आपका जो ग्लाइकोलाइटिक एंड प्रोडक्ट है दैट इज पायरुवेट वो बिल्डअप होना शुरू हो जाएगा. इट विल नेवर बी कन्वर्टेड इनटू एसिटाइल कोए, जो कि कनेक्टिंग लिंक है बिटवीन ग्लाइकोलाइसिस एंड क्रेब्स साइकिल. तो ऑब्वियसली बात है कि जब आपका पायरुवेट डिहाइड्रोजनेज वर्क ही नहीं कर रहा है बिकॉज ऑफ द डेफिशिएंसी ऑफ थायमिन, तो पायरुवेट जो है वो बिल्डअप होगा और कन्वर्ट होगा नहीं एसिटाइल कोए में. तो ऑब्वियसली इसका आंसर होगा ऑप्शन सी, दैट इज थायमिन डेफिशिएंसी. What type of chemical reaction is involved in conversion of isocitrate to alpha ketoglutarate? Carboxylation, oxidative decarboxylation, reducing decarboxylation, or oxido reduction. If you see, isocitrate in 2 steps it is being converted into alpha ketoglutarate. पहले स्टेप में ऑक्सीडेशन होता है आइसोसाइट्रेट का, जो कि कन्वर्ट कर देता है इसको ऑक्सैलोसक्सिनेट में, और दूसरे स्टेप्स में डीकार्बोक्सिलेशन स्टेप्स के थ्रू इट इज बीइंग कन्वर्टेड इनटू अल्फा कीटोग्लूटारेट. तो 2 स्टेप्स में फर्स्ट ऑक्सीडेशन, उसके बाद में फॉलोड बाय डीकार्बोक्सिलेशन. ऑक्सीडेटिव डीकार्बोक्सिलेशन एक्चुअली इट कन्वर्ट आइसोसाइट्रेट टू अल्फा कीटोग्लूटारेट. तो ऑप्शन बी हैज टू बी द करेक्ट आंसर फॉर दिस क्वेश्चन. Would be the effect on the net reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase, which is GAPDH, if phosphate was replaced by arsenate? Rate of the reaction will increase, rate will be decreased, no effect on reaction rate, or uncoupling of phosphorylation. GPEA-PADH जो enzyme है glyceraldehyde 3-phosphate dehydrogenase, what it does is it converts glyceraldehyde 3-phosphate into 1,3-biphosphoglycerate. When we say 1,3-biphosphoglycerate, it simply means एक फर्स्ट पोजीशन पे जो कार्बन है आपका, उस पे आपका एक फॉस्फेट ग्रुप एक्स्ट्रा ऐड कर देता है ये एंजाइम. आर्सेनेट जो है वो एक नेचुरली माइमिक करता है फॉस्फेट ग्रुप को, यानी कि अगर फॉस्फेट और आर्सेनेट प्रेजेंट होगा तो कोई भी एंजाइम इंस्टेड ऑफ पुटिंग फॉस्फेट आर्सेनेट को भी एक चॉइस के तौर पे ले सकता है, एंड इट कैन बी ग्राफ्टेड ओवर द इट्स प्रिकर्सर मॉलिक्यूल्स. इस केस में अगर हम बात करें तो अगर आर्सेनेट प्रेजेंट होगा इंस्टेड ऑफ फॉस्फेट तो आर्सेनेट आपका जाके फर्स्ट पोजीशन पे लग जाएगा, यानी कि जो फॉस्फोरिलेशन की जो कपलिंग है वो नहीं हो पाएगी. इट वुड बी अनकपलिंग ऑफ फॉस्फोरिलेशन. इंटरेस्टिंगली अगर सपोजिटली आर्सेनेट लग भी जाता है तो जो कंपाउंड बनता है वो हाईली अनस्टेबल होता है, अपने आप से स्पॉन्टेनियसली डिग्रेड होगा. आप एंजाइम्स की जरूरत नहीं पड़ेगी नेक्स्ट स्टेप में उस कंपाउंड को डिग्रेड करने के लिए. तो इस केस में द आंसर वुड बी ऑप्शन डी, दैट इज अनकपलिंग ऑफ फॉस्फोरिलेशन इज ड्यू टू द कंपटीशन proposed by the arsenic molecule. In glycolysis, pyruvate dehydrogenase is subjected to feedback inhibition by its products. The chemical compounds which might be involved in the process are listed below: A. NADH, B. FAD, C. acetyl-CoA, and D. acetaldehyde. Now, which one of the following combination of above chemical compounds is involved in the feedback inhibition of pyruvate dehydrogenase? As you can see in the figure, pyruvate dehydrogenase can positively regulate by pyruvate and negatively regulated by NADH, acetyl-CoA, and ATP. So here option D, that means A, which is NADH, and C, which is acetyl-CoA, will be the one which is going to inhibit the pyruvate dehydrogenase through feedback inhibition. Coenzymes involved in the formation of acetyl-CoA from pyruvates are thymine pyrophosphate, lipoic acid and FAD, pyridoxal phosphate, biotin and FAD, vitamin B12, folic acid and vitamin C, NADH, lipoic acid and vitamin E. During the end stage of glycolysis, pyruvate is converted to acetyl-CoA by an enzyme called pyruvate dehydrogenase complex. This complex requires coenzyme ASH, NAD as a substrate, and TPP, lipoid, and FAD as an coenzyme in sequential manner. So here the option A has to be the correct answer, that thymine pyrophosphate, lipoic acid, and FAD are the common coenzymes which are used by the pyruvate dehydrogenase complex along with CoA-SH and NAD. Which cofactor is crucial in catalyzing enzymatic conversion of pyruvate to lactate? NAD, NADH, FAD, or FADH? If you see it in a diagram, that lactate dehydrogenase or LDH, which catalyze the conversion of pyruvate to lactate using NADH as a cofactor. So option B is the correct answer. Which of the following pair of amino acids are both glucogenic and ketogenic in nature? Alanine and lysine, lysine and leucine, isoleucine and phenylalanine, or aspartate and lysine. If you can see it in the figure, you can figure it out that lysine and leucine, they are the pure ketogenic amino acids. While there are 5 sets of amino acids which are both glucogenic and ketogenic. They are isoleucine, leucine, phenylalanine, tryptophan, and tyrosine. So for the given question, the answer has to be the option C, that isoleucine and phenylalanine are both glucogenic and ketogenic in nature. The citric acid cycle in respiration is. So if we look at the diagram, we can find out that one round of citric acid cycle or Krebs cycle, the output product would be 3 NADH, 1 FADH2, 1 GTP, or 2 CO2. So option A is the correct answer. The only enzyme belonging to Krebs cycle that is attached to inner mitochondrial membrane, that's very easy. Succinate dehydrogenase, also known as respiratory complex 2, is found embedded in inner mitochondrial membrane of eukaryotes. It is only enzyme which is common to Krebs cycle and electron transport chain. During electron transport, protons are pumped out of the mitochondria at each of the major sites except for complex 2, complex 4, complex 3, or complex 1. The energetically downhill movement of electron through the electron transport chain causes the pumping of hydrogen ion or protons into the intermembrane spaces by first, third, and fourth complex. Second complex is failed to transfer the protons. So here the option A is the correct answer. Uncouplers like DNP or thermogenin uncouple electron transport chain and oxidative phosphorylation by inhibiting cytochrome oxidase, blocking electron transfer, dissipating the proton gradient, or blocking ATP-ADP translocase. Uncouplers of oxidative phosphorylation actually disconnect the activity of electron transport chain and the ATP synthesis. Actually, they allow the reverse migration of the protons which have been generated by the electron transport chain activity. Now the protons are get back to the matrix, and now there is no proton gradient is being left. Since there is no proton gradient is there, so no driving force is there to synthesize the ATP, and hence ATP synthesis is being compromised. So here the uncouplers like DNP or thermogenin basically uncouple by dissipating the proton gradient. Ribulose 5-phosphate epimerase is involved in glycolysis, TCA cycle, pentose phosphate pathway, or fatty acid. L-ribulose 5-phosphate 4-epimerase, it catalyzes a interconversion of ribulose 5-phosphate and xylulose 5-phosphate in the oxidative phase of the pentose phosphate pathway, or also known as hexose monophosphate shunt, that is C. Glycogen phosphorylase exists in two forms in skeletal muscle. The active form of phosphorylase A is generated from phosphorylase B by reversible dimerization of phosphorylase B triggered by calcium ion, proteolytic cleavage of a decapeptide from the N-terminus of phosphorylase B, protonation of the active site histidine residue by a decrease in intracellular pH or ATP dependent phosphorylation of a specific serine residue on each subunit. In liver and muscles, glycogen phosphorylase enzyme is allosterically regulated through phosphorylation step. Active form of a phosphorylation type A is converted to inactive phosphorylation beta type by the reversible phosphorylation of a serine 14 residue. So here the option D has to be the correct answer, that ATP dependent phosphorylation of a specific serine residue on each subunit is basically responsible for the conversion of active form of a phosphorylase A from phosphorylase B. Process of production of glucose from amino acids and the glycerol portion of the fats is known as gluconeogenesis, glycogenolysis, glycerogenesis or glucosis. Gluconeogenesis is a metabolic pathway by which glucose is formed from the non-carbohydrate sources like lactate, amino acids or glycerol. So option A is the correct answer. Mammalian liver can carry out gluconeogenesis using starting material known as. ध्यान से देखिए, जो आपका स्टार्टिंग मटेरियल ग्लूकोनियोजेनेसिस के होते हैं वो जनरली 3 कार्बन कंपाउंड्स होते हैं लाइक लैक्टेट, पायरुवेट, ग्लिसरॉल, कुछ अमाइनो एसिड्स. लीवर में ग्लूकोनियोजेनेसिस कैन बी इनिशिएटेड बाय यूजिंग ऑक्सैलोएसीटेट एज एन स्टार्टिंग मटेरियल. तो इसमें ऑप्शन ऑब्वियसली होगा ऑप्शन ए. आगे चलते हैं. The biochemical products obtained after hydrolysis of a glycolipids are sugar, fatty acid, phosphoric acid, sugar, fatty acid, nitrogenous base, sugar, fatty acid, glycerol, sugar, fatty acid, sphingosine. इसका ऑप्शन बहुत ही आंसर क्लियर है. ऑप्शन सी हैज टू बी करेक्ट. क्यों? क्योंकि अगर आप ध्यान से देखिए ग्लाइकोलिपिड्स वर्ड है, ग्लाइको मतलब शुगर, एक आना ही बनता है, तो इसीलिए आप देख सकते हैं कि चार ऑप्शंस पे शुगर है. बचता है लिपिड्स. लिपिड्स कैसे बनते हैं? अगर आप ध्यान से देखिए लिपिड्स की जो सिंथेसिस होती है उसके लिए जो प्रिकर्सर्स होते हैं वो होता है एक तो ग्लिसरॉल मॉलिक्यूल और फ्री फैटी एसिड्स. तो यानी कि फ्री फैटी एसिड्स और ग्लिसरॉल इज कमिंग बिकॉज ऑफ द लिपिड्स एंड ग्लाइको वर्ड्स की वजह से जो शुगर आएगा. तो शुगर, फैटी एसिड एंड ग्लिसरॉल दे आर द main precursors or they will be the degraded products once the glycolipids are being hydrolyzed. So option C has to be the correct answer. Principal regulation point in the biosynthesis of fatty acid is acetyl CoA carboxylase, beta ketoacyl ACP synthase, citrate dehydrogenase और बीटा लैक्टामेज. अगर हम ध्यान से देखें तो जो सैचुरेटेड फैटी एसिड सिंथेसिस है उसमें एसिटाइल कोए कार्बोक्सिलेज जो है दैट इज द मोस्ट कमिटेड स्टेप, मोस्ट रेगुलेटरी चेक पॉइंट है और जिसको के फॉस्फोरिलेशन के थ्रू या एलोस्टेरिक रेगुलेशंस के थ्रू कंट्रोल किया जाता है. और इसीलिए इस क्वेश्चन का आंसर होगा एसिटाइल कोए कार्बोक्सिलेज विच इज द मोस्ट इंपॉर्टेंट स्टेप और एंजाइम जो कि रेगुलेशन में सब्जेक्ट किया जाता है. तो ऑप्शन ए हैज टू बी द करेक्ट आंसर. From the given intermediates in the fat metabolism, correct intermediate series is. So there are 4 options are given and we have to arrange the intermediate as per they happen during the beta oxidation of free fatty acid. So let's start. If you see it in the diagram, once we start with the fatty acyl CoA, after an oxidation step, it is being converted into the transenoyl CoA having a double bond. Now across this double bond, there is an addition of water molecule takes place. As per the Setzeff rule, the OH is being added at the beta position, which after one round of oxidation is being converted into a keto group and the compound will be ketoacyl CoA. Now, a thiolysis steps actually cleave this molecule at the beta position and led to the release of an acetyl CoA. And now, a free fatty acid is formed, which is a 2 carbon short from the beginning. This molecule is again ready for the next round of beta oxidation and it keeps on happening in a spiral case manner till the last acetyl CoA is left. So the order of intermediate would be fatty acyl CoA led to the formation of transenoyl CoA having a double bond, and now it led to the formation of beta hydroxyacyl CoA after the addition of the water, and last the ketoacyl CoA which is being degraded through the thiolysis step into the acetyl CoA. So option A is the correct answer. Lecithin are composed of glycerol plus fatty acid plus phosphoric acid plus serine, glycerol plus fatty acid plus phosphoric acid plus ethanolamine, glycerol plus fatty acid plus phosphoric acid plus choline, or glycerol plus fatty acid plus phosphoric acid plus betaine. Lecithin are the phosphoglycerides made up of condensation of glycerol with at least one phosphorylated fatty acid, that is phosphoric acid plus choline and two fatty acids. So here the option C has to be the correct answer. At room temperature, that is 25 degrees Celsius, an unsaturated fatty acid will have solid consistency, waxy consistency, liquid consistency, or cannot be predicted. Due to the presence of double or triple bonds, unsaturated fatty acids are liquid at room temperature. So option C is the correct answer. RNA lacks which of the following? O-glycosidic bond, N-glycosidic bond, purines, or ester bond. In nucleic acid, the sugar is linked to the nitrogenous base, that is purine or pyrimidine, with their first carbon, or we also call it as anomeric carbon, with the help of N-glycosidic bond. While at the other end of the sugar, it is connected with another sugar with the phosphodiester bond. So option A is the correct answer, that RNA lacks basically O-glycosidic bond. N-glycosidic bond is present between a sugar and the nitrogenous base. It has definitely purines and pyrimidines, and at the other end it has a phosphodiester bond, while O-glycosidic bond is definitely absent. Select the true statement about phosphodiester linkage. 3' phosphate group of one nucleotide unit is joined to the 5' hydroxyl group of the next nucleotide. 5' phosphate group of one nucleotide unit is joined to the 3' hydroxyl group of the next nucleotide. 5' phosphate group of one nucleotide unit is joined to the 5' hydroxyl of the next nucleotide, or 3' phosphate of one nucleotide unit is joined to the 3' hydroxyl of the next nucleotide. Phosphodiester linkage is created when the 5' end of one nucleotide having phosphate group react with the 3' hydroxyl of another nucleotide which is a part of the existing strand. So here the option B has to be the correct answer. DHFR or dihydrofolate reductase, it catalyzes the conversion of. As we all know, DHFR is an important enzyme of de novo purine or thymidylate synthesis. It converts the dihydrofolate to tetrahydrofolate using NADPH as an electron donor. So here the option B would be the correct answer. The DHFR, it catalyzes the conversion of dihydrofolic acid to THF using the NADPH as an electron donor. Which of the following statement about the salvage pathway for the synthesis of purine nucleotide is correct? Increased PRPP level in the cell inactivate HGPRT. Decreased PRPP level in cell lead to the activation of HGPRT. Increased PRPP level in the cell led to the activation of HGPRT, or purine nucleotide competitively inhibits HGPRT. HGPRT basically catalyzes salvage pathway of purine bases using PRPP as a substrate. This PRPP dependent reaction by HGPRT actually gets increased as the concentration of PRPP increases, while deficiency of this enzyme resulted in the accumulation of its substrate, that is hypoxanthine and guanine and even PRPP. So here if we see, the increased PRPP level in the cells led to the increase of the HGPRT, that is option C. A drug which prevents uric acid synthesis by inhibiting the enzyme xanthine oxidase is aspirin, allopurinol, colchicine, or probenecid. Allopurinol, which is also known as xyloprene, actually it inhibits the enzyme xanthine oxidase, which actually synthesizes uric acid from hypoxanthine in purine metabolism. So option B is the correct answer. As far as vitamins are concerned, याद रखिए एक टेबल बना लीजिए, इंपॉर्टेंट उनके साइंटिफिक नेम, उनके केमिकल स्ट्रक्चर्स की अगर कोई प्रॉपर्टीज आपको आती है, एंड इंपॉर्टेंट उनसे जो डिजीज होती है, राइट? ये चीजों को अगर आप रेफर कर लेंगे तो आपके कई क्वेश्चंस इसमें से बन जाएंगे. ओके. The vitamins having the highest daily requirement among the following is thiamine, riboflavin, pyridoxine, or ascorbic acid. Among different vitamins, vitamin C can be taken in without having dietary complication. As far as males are concerned, they can take up to 75 mg, while females are required much more ascorbic acid, that is around 90 mg. So option D is the correct answer. Pyridoxal phosphate is the functional form of vitamin B2, vitamin B6, vitamin B3, or vitamin B12. In its active form, vitamin B6 comprise three organic compounds: pyridoxal, pyridoxamine, and pyridoxine. So here the option B is the correct answer, that vitamin B6 is the functional form of pyridoxal phosphate. Hence the coenzymes in list 1 items serving as a transient carrier of specific atoms or function group in list 2. So list 1 is having 4 coenzymes, and you have to find out that to which group or functional group it is going to react. So let's start. Coenzyme A. Coenzyme A is basically has a terminal cysteine residue or SH group to which an acyl group is tethered. Generally, coenzyme A is also known as acyl group carrier. So A has to be correctly matched with the 4 acyl groups. Now FAD. FAD is often found as a redox cofactor and exists in two redox states: FAD, which is an oxidized state, and once it accepts 2 hydrogen atoms, it becomes FADH2, which is a reduced state. So FADB has to be connected with third. Now pyridoxal phosphate. Pyridoxal phosphate is a coenzyme who is having a aldehydic group and react with the amino group of any amino acids or any other molecule which is having NH2 group and forms the shift linkage. So C has to be connected with 1. No. We have to find out to which group it reacts, not that the group it contains. So pyridoxal phosphate definitely has an aldehyde group, but it reacts with amino group. So C has to be connected with 2. Now thymine pyrophosphate, it has a thiazole ring which is having an acidic proton and it can bind with the aldehyde group. So D has to be correctly matched with the first aldehyde group because it reacts with aldehyde group. So option A is connected with 4, B connected with third, C connected with second, and D connected with 1. That is option A. Which vitamin helps in clotting of the blood? Vitamin C, vitamin K, vitamin A, or vitamin E? That's very simple. Vitamin K is required by the body for blood clotting, and hence it helps in healing of the wound. So option B would be the correct answer. A deficiency of folate led to beriberi, megaloblastic anemia, pernicious anemia, or hypochromic microcystic anemia. Low levels of folic acid can cause megaloblastic anemia, in which the RBC precursors are way larger than the normal cells in the bone marrow. So option B is correct. Anemia is caused due to the deficiency of cobalamin, pyridoxine, folic acid, or niacin. Most common cause of a megaloblastic anemia are due to the deficiency of either cobalamin, which is vitamin B12, or folate, which is vitamin B9. तो इसी क्वेश्चन में अगर हम ध्यान से देखें तो ऑप्शन ए एंड सी बोथ आर करेक्ट, तो इसीलिए वी विल बी लाइक ऑप्शन सी हम यहाँ पे मान रहे हैं फोलिक एसिड, जो कि एग्जाम में दिया हुआ है, क्वेश्चन पेपर का जो आंसर किया गया है ऑफिशियल, उसमें फोलिक एसिड दिया हुआ है, बट कोबालामिन कुड आल्सो बी अ करेक्ट आंसर. Vitamin B12 is absorbed primarily in the jejunum, duodenum, stomach, or ileum. Vitamin B12 or cobalamin is a water-soluble vitamin found in the animal-based foods. It is absorbed in the last part of the small intestine, which is also called as ileum. So option D is the correct answer. Transketolase activity is affected in biotin deficiency, ascorbic acid deficiency, PABA deficiency, or thymine deficiency. Transketolase is basically a thymine pyrophosphate dependent enzyme, and its activity is being affected by the thymine deficiency. there are two more enzymes whose activity is also get affected by the thymine deficiency. Those are pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase of the Krebs cycle. Excessive intake of which of the following element cause mottled teeth and enlarged bone? Magnesium, fluorine, phosphate or calcium. Ingestion of excess fluoride may cause the bone enlargement or dental fluorosis. So option B must be the correct answer. बायोकेमिस्ट्री की बात हो और एंजाइमोलॉजी या थर्मोडायनेमिक्स ना आए, ऐसा होता नहीं है. इंटरेस्टिंगली थर्मोडायनेमिक्स पे क्वेश्चंस जो हैं कम आए हैं एमएससी बायोकेमिस्ट्री एंट्रेंसेस में, बट एंजाइमोलॉजी में एंजाइम्स की जनरल प्रॉपर्टीज और एंजाइम इनहिबिशंस उनको अच्छे से करके जाइएगा. देखिए कुछ क्वेश्चंस को डिस्कस कर लेते हैं. Which of the following statement is true about enzymes? Enzyme increases the equilibrium constant of the reaction they catalyze? No. Enzyme do not alter the overall change in the free energy for a reaction? Yes. Enzyme do not influence the rate constant of the forward reaction they catalyze? No. Enzyme speed up the reaction by increasing the activation energy? No. They actually decrease the activation energy. Basically, we should remember what is do regarding the enzyme, what is don't regarding the enzyme. Actually, enzyme fasten up the reaction and actually they influence the rate constant of the forward reaction. Second, what they do, they actually lower down the activation energy in order to fasten up the reaction. They never disturb the equilibrium constant, they never disturb the free energy change, they never disturb the enthalpy, or they also never disturb the entropy of a reaction. So here the option B is the correct answer, that enzymes do not alter the overall change in the free energy for a reaction. Proteolytic enzymes synthesized as large inactive precursors are known as, again one of the most simple questions. So as you are seeing that MSc Biochemistry questions are not so difficult which you cannot crack. So please trust yourself and start appearing. Now let's get back to the question. Proteolytic enzymes are synthesized as large inactive precursor which is also known as proenzyme or zymogen. For example, pepsinogen is an zymogen which remain inactive due to the presence of 44 amino acid long masking peptide. Upon removing this peptide, it becomes an active form of pepsin. So, proteolytic enzymes synthesized as large inactive precursors are known as proenzyme or zymogen. Which of the following protein is not synthesized as zymogen or proproteins? Pepsin, lysozyme, collagen, trypsin. Lysozyme is an enzyme which is released directly in functional state, while pepsin, collagen and trypsin are released as pepsinogen, procollagen and trypsinogen, which are the inactive format of all these three proteins. So option B is the correct answer. Which one below can break a peptide bond enzymatically? Papain, myosin, cyanogen bromide or performic acid. Papain is a protease which can break peptide bond by using its catalytic triad with a deprotonated cysteine at its active site. The enzyme that hydrolyzes starch into the maltose is glucoamylase, alpha amylase, beta amylase or glucotransferase. Beta amylase catalyzes the hydrolysis of starch into the maltose by hydrolysis of alpha 1,4 glycosidic bond. So option C is the correct answer. Chymotrypsin is specific for the peptide bonds containing. Chymotrypsin is basically a typical serine protease which preferentially cleave a peptide bond in which the N-terminal amino acid to the bond is a large and uncharged aromatic amino acid which could be phenylalanine, tryptophan, tyrosine or even leucine. So here the option A is the correct answer. In the enzyme catalyzed biosynthetic pathway shown below, what will be the effect on the substrate A, B, C and D if the enzyme E2 is inactivated? That's very simple. As we can see, if E2 is deficient, then downstream product C and D will not form, but B can still form because E1 is still active. So option C is correct, that V will be produced but not the C or D. Isoenzymes are chemically, immunologically and electrophoretically different forms of an enzyme. Different forms of an enzyme similar in all properties, catalyzing different reactions or having the same quaternary structure like the enzymes. Isoenzymes are those enzymes which catalyze the same reactions but occur in a number of different forms. All these different forms are different in terms of either chemically or immunologically or even electrophoretically migrations. Multi-enzyme systems include fatty acyl synthetases, hexokinases, phosphatases, or lysosomes. Fatty acyl CoA synthetase is a multi-subunit enzyme which catalyzes the ATP-dependent formation of fatty acyl CoA from free fatty acid. This step actually prepares fatty acid to yield ATP by using beta oxidation process. There is another enzyme which actually associated with fatty acid anabolism, that is fatty acid synthase. This enzyme is also a multi-subunit enzyme having 6 enzyme activities. So here the enzyme fatty acyl synthetase is an answer that it is a multi-subunit enzyme which is involved with fatty acid catabolism. Enzyme with highest turnover number would be lysozyme, G3PDH, carbonic anhydrase or LDH. The number of substrate molecules converted into the product per minute by an enzyme is called as turnover number. Higher the turnover number, more efficient enzyme would be. Catalase has the highest turnover number where one molecule of catalase may convert over 2.8 million molecules of hydrogen peroxide to water. Carbonic anhydrase is the another incredibly fast enzyme with a kcat of 10 to the power 6 reactions per second. So carbonic anhydrase along with catalase, they are the two best known enzymes with the highest turnover number. An enzyme E has a kcat value of 600 per second. Now we have to calculate the Km value when reaction proceeds at a rate of 9.6 micromolar per second and substrate concentration is 40 micromolar. The total given concentration of enzyme is 0.02 micromolar. As we know, the formula of kcat is Vmax by total enzyme concentration. So if we place the value of kcat and total enzyme concentration which is being given, then we can get the value of Vmax. Now if we place the value of Vmax in a classical Michaelis-Menten equation, then we know that we have the values of V, that is instantaneous velocity, Vmax, S, and we have to find out the value of Km. Km, after solving the values, then we can get the value as 10 micromolar. So option C has to be the correct answer. One international unit, that is IU of vitamin D, is defined as the biological activity of 0.025 microgram of cholecalciferol, 0.025 microgram of 7-dehydrocholecalciferol, 0.025 microgram of ergosterol or 0.025 microgram of ergocalciferol. If you see it in the figure, the mass equivalent of 1 IU for the various types of vitamins are given. So 1 IU of vitamin D is the biological equivalent of 0.025 microgram cholecalciferol, which is option A. Enzymes are also referred to as zymogens, enzymes that are highly specific like antibodies, enzymes that hydrolyze antibody or antibodies with the catalytic activities. Enzymes or catalytic monoclonal antibodies are the antibodies having capability to bind transition state and they facilitate the product formation just like enzyme. So option D is the correct answer, that enzymes are antibodies that have the catalytic activities. Statins, a popular class of drug, is known to inhibit HMG-CoA oxidase, GADPH, HMG-CoA reductase or pyruvate decarboxylase. Statin actually blocks an enzyme called HMG-CoA reductase that is involved in the synthesis of an steroid intermediate called mevalonate. So option C is the correct answer. Which of the following property is not exhibited by an allosteric enzyme? So इसमें से 4 प्रॉपर्टी में से we have to find out which is not a characteristic properties of an allosteric enzyme. Sigmoidal substrate binding curve. Definitely, sigmoidal substrate binding curve is an hallmark feature of an allosteric enzyme. Allosteric enzymes की क्या-क्या properties होती हैं? They are multimeric in nature. That means they are quaternary structure. They have a typical substrate binding curve. That means sigmoidal substrate binding curve, unlike of a typical hyperbolic curve shown by the Michaelis-Menten enzyme. Then they have a cooperativity, and now they have a two different sites. One is a regulatory site and one is substrate binding site. So one thing is very clear as far as allosteric enzyme, that no allosteric enzyme can be a typical Michaelis-Menten enzyme. So option B is the correct answer, that adherence to Henry-Michaelis-Menten equation is not a characteristic of allosteric enzyme. In fact, while explaining the kinetics, they have assumed that no enzyme should be an allosteric enzyme if they have to be solved by using kinetics. So option B is the correct answer. लाइनवीवर वर्क प्लॉट इज ऑल्सो नोन एज एलबी प्लॉट जो होता है, जो कि आपका माइकेलिस मेंटन का एक अडेप्टेशन है, उसको हम डबल रेसिप्रोकल प्लॉट भी बोलते हैं, जिसमें कि एक्स एक्सिस पे आपका जो होता है वो 1/s होता है और जो आपका वाई एक्सिस है उसमें 1/v होता है। और दोनों ये जो प्लॉट है ये बड़ा फेमस है बिकॉज इसकी मदद से हम km और vmax की जो एक्यूरेट वैल्यूज हैं वो उनको हम निकाल सकते हैं। तो एलबी प्लॉट जो है दैट इज ऑल्सो नोन एज double reciprocal plot, that is option B. In a competitive enzyme reaction, Km and Vmax decreases, Km and Vmax increases, Km increases and Vmax does not show any change. Km shows no change while Vmax decreases. Basically, competitive inhibitors, they compete with the substrate at the active site and therefore they increase the Km but the Vmax remain unaffected. So option C is the correct answer. A certain small molecule inhibits an enzyme by binding only to the enzyme substrate complex, or in other words, by binding only the substrate binds the enzyme. This particular inhibition is a typical of which class of inhibition? First of all, you should know what are the three types of inhibition: competitive, uncompetitive, non-competitive, and what is their pattern. Competitive inhibition कब होता है? Competitive inhibition is there when a inhibitor competes for the active sites. So that cannot be the answer here. Non-competitive inhibition कब होता है? When a substrate binding is being impaired due to the binding of inhibitor at some distant site. So this can also not be the answer because substrate is binding here. Now, in the case of uncompetitive inhibition, that one of the hallmark feature of uncompetitive inhibition is that it only gets activated when a substrate binds to the enzyme. That means the target of an uncompetitive inhibitor is the ES complex. So here the option D is the correct answer. जब भी थर्मोडायनामिक्स की बात आएगी तो फ्री एनर्जी जो चेंज है उसके साइन कन्वेंशंस को अच्छे से करके जाइएगा। स्पॉन्टेनिटी के जो सिग्नेचर्स हैं उनको अच्छे से पढ़ लीजिएगा, आपके क्वेश्चंस हो जाएंगे। In a biochemical reaction, the thermodynamic parameter that reaches a minimum is, that's again very easy, enthalpy, entropy, free energy, enthalpy and entropy both. As we know that delta G is an important indicator of spontaneity and extent of any chemical reaction. In a spontaneous change, any reaction proceeds towards those directions which led to the minimum delta G or the negative sign of delta G. Hence, option C would be the correct answer. Which of the following acts as an ATP buffer in human body? AMP, adenosine, phosphokeratin or phosphatase. Keratin phosphate, it serves as an energy buffer which helps in maintaining a constant concentration of ATP in muscles during sudden need. It helps in those conditions when suddenly cellular ATP are exhausted due to the requirement and hormonal induced catabolic pathway is yet to start. Breaking of keratin phosphate bond releases 43.3 kilojoule per mole energy, which is considerably much more than the energy which we get after the breakdown of ATP. Hence, at times this energy which is released due to the breakdown of phosphokeratin is used to recharge the ATP. So phosphokeratin is a known energy buffer in our body. especially in muscle cells. Okay, टेक्निक्स में वैसे तो सारी ही टेक्निक्स इंपॉर्टेंट होती हैं, बट अगर आपको टॉप 3 टेक्निक्स चुननी है तो आपकी स्पेक्ट्रोस्कोपी पे क्वेश्चंस हैं, स्पेशली न्यूमेरिकल्स, और इसके अलावा आपके इलेक्ट्रोफोरेसिस एंड आइसोइलेक्ट्रिक फोकसिंग, इन पे बहुत क्वेश्चन रिपीट हुए हैं, जैसा कि अभी हम देखेंगे भी, ठीक है। Specific functional group in an amino acid undergoes specific chemical reaction producing a typical color that helps in the detection of the relevant amino acid. Match the amino acid in list 1 to their specific test with list 2. So list 1 outlines the name of the test and we have to correctly match that which amino acid can be characterized by using these tests. So let's start. Hopkins color test. Hopkins color test is used to find out the indole ring. Upon reaction, indole ring gives the violet color. Now, indole ring is present in which amino acid? That is tryptophan. So A has to be connected with second, that is tryptophan. Polys test. Polys test is used for the quantitative detection of tyrosine and histidine. So, obviously, by using Polys test, we cannot differentiate among tyrosine and histidine. But in order to differentiate, we can use Millen's test because Millen's test is very specific for tyrosine. So D, that is Millen test, can be used for tyrosine, and indirectly poly test can be used for the histidine. That means B is connected with third. Leftover nitroprusside, that can be used to characterize the cysteine at alkaline pH. So A will be connected with 2, B will be connected with 3, C will be connected with 4, while D will be connected with first, that is option A. The extinction coefficient of a protein at 280 nanometer is 20 millimolar per centimeter. The absorbance of the protein solution at 280 nanometer appropriately diluted measured in a cuvette with a path length of 1 centimeter is 100. What is the concentration of the protein solution? If we solve this question, then we will find that the Lambert-Beer law is very easy to use in this case. Here we are being given the absorbance, that is 100, the molar extinction coefficient, that is 20 millimolar inverse per centimeter inverse, and the path length is 1 centimeter. If we solve it for the concentration comes out to be 5 millimolar, that is option A. Which one of the following is not a protein estimation method? Bistinchonic acid method, biuret method, Barford method, or Lowry method? Barford method is used to detect the presence of monosaccharide. It is based on the reduction of Cu2 plus state to Cu1 plus state, which forms the brick-red PPT. So now the Barford method is basically used for the quantitation of monosaccharide, while rest of the other methods are used for the estimation of protein. So option C is the correct answer. Absorbance of a DNA solution can be measured using fluorimeter, spectrophotometer, thermometer, or radiometer. The absorbance of a DNA sample is measured at 260 nanometer on a spectrophotometer using UV transparent cuvettes. Which chemical you will use for quantifying DNA? Densyl chloride, diphenylamine, orsinol, or ninhydrin? Diphenylamine is used for the quantitation of DNA, where under acidic conditions DNA react with diphenylamine dye and form bluish green complex. Now the color can be measured at 595 nanometer. Always remember that this method is non-specific and nowadays not is much used. So option B is the correct answer. Match the techniques in list 1 with the application given in the list 2. So list 1 outlines the different techniques, while list 2 outlines their basically specific uses. A. Salting out. B. Ultracentrifugation. C. Dialysis. While D. Isoelectric focussing. See, salting out is a method of protein precipitation at high salt concentration. That means A has to be connected with 2, protein precipitation. While dialysis, which is C, is a method which is used to remove these salts from protein precipitation. That means C has to be connected with 4, removal of low molecular weight, that means salt impurities. So we have to find out in which option A is connected with second while C is connected with fourth. If you see the option A is connected with second while C is connected with fourth. That has to be the correct answer. Ultrafiltrate centrifugation, as we know, is used to determine the sedimentation coefficient, while isoelectric focusing is also used to determine the PI. The salt which is most commonly used for salting out effect during extraction of protein is, that's very easy. Ammonium sulfate is the most commonly used protein purification method at the high salt concentration. That is also known as salting out conditions. So option B is the correct answer. An example of non-ionic detergent is triton X100, benzalkonium chloride, sodium dodecyl sulfate, or CETAB. Basically, detergent which do not have anionic or cationic charge on them, they are called as non-ionic detergent, like triton X100. It is used for cell lysis. Remember, SDS, which is sodium dodecyl sulfate, is an anionic detergent, while CETAB is an example of cationic detergent. So here option A is the correct answer. Denaturation of a protein or a nucleic acid can be studied by SDS patch, isoelectric focusing, स्पेक्ट्रोफोटोमेट्री और जेल फिल्ट्रेशन। बड़ा इंटरेस्टिंग सा क्वेश्चन है। अगर आप ध्यान से देखें, जो डीनेचुरेशन स्टडीज हैं जनरली दे आर बीइंग डन बाय द स्पेक्ट्रोस्कोपिक मेथड्स। यूवी विजिबल आप ले लीजिए, सीडी आप लीजिए, सर्कुलर ड्राइकोइज्म, फ्लोरोसेंस बेस ले लीजिए, आईआर, एनएमआर आपकी टेक्निक्स हैं, लाइट स्कैटरिंग टेक्निक्स हैं, ये सबको आप यूज कर सकते हैं डीनेचुरेशन स्टडी करने के लिए प्रोटीन्स में और न्यूक्लिक एसिड में। फोल्डिंग अनफोल्डिंग दोनों तरह की स्टडीज के लिए जनरली स्पेक्ट्रोफोटोमेट्रिक स्टडीज जो होती हैं वो ज्यादा एप्ट मानी जाती हैं। दे हैव अ मोर वर्सेटिलिटी, और इसीलिए ऑप्शन जो सी है दैट हैज टू बी द करेक्ट आंसर। दिस पास द माइग्रेशन ऑफ अ प्रोटीन इज इफेक्टेड बाय, बेसिकली होता क्या है कि इन एसडीएस पास द माइग्रेशन रेट जो होता है वो डिक्टेट होता है उसके साइज के या शेप के हिसाब से, और विच इज अल्टीमेटली अ रिफ्लेक्टेंस ऑफ अ मास। तो अल्टीमेटली जो साइज ऑफ द प्रोटीन है वो इफेक्ट करता है बेसिकली माइग्रेशन ऑफ अ प्रोटीन इन एसडीएस पास। तो ऑप्शन बी should be the correct answer in this. Glycerol is added to a protein samples before loading them on the patch. What is the role of a glycerol? Glycerol basically प्रोटीन वर्ल्ड में बहुत ज्यादा यूज होता है, बहुत सारे रीजन है। ग्लिसरॉल एक क्रायोप्रोटेक्टेंट है, तो जो थॉइंग वगैरह में यूज होता है, और ग्लिसरॉल के और भी यूज हैं। बट एसडीएस पास में या नॉर्मल पास में होता क्या है, ग्लिसरॉल एक जो है वो डेंसिटी प्रोवाइड करता है सैंपल्स को। उसकी वजह से क्या होता है, जो सैंपल्स हैं वो बड़े आराम से आपके जो वेल्स बनाए हैं उनमें चले जाते हैं। अदरवाइज जो आपका अगर ग्लिसरॉल नहीं पड़ेगा होगा तो डेंस नहीं होगा सैंपल, और वो जो है फ्लोट करना लग जाएगा आपके बफर्स में, इलेक्ट्रोलाइट बफर्स में। तो इसीलिए आपको याद रखना है कि क्या नाम है, ग्लिसरॉल इज एडेड टू द प्रोटीन सैंपल बिफोर लोडिंग देम ऑन अ पार्ट। तो रोल ऑफ अ ग्लिसरॉल इज टू प्रोवाइड डेंसिटी टू द प्रोटीन सैंपल। Different form of a plasmid on an agarose gel after electrophoresis may be in which of the following sequence in decreasing molecular weight? First of all, one should know that in electrophoresis, higher molecular weight or open conformation observe retarded migration than the compact or low molecular weight conformations. So plasmid DNA, which can exist in three conformations, that is supercoiled, which is most compact, open circular, or linear one, and each one can migrate at a different rates for the same overall size. That means if we take the plasmid in these three conformations but have the same size, then supercoiled DNA being compact may run faster and than open circular. Linear DNA observe less friction than open circular conformation. Hence, the order will be the supercoiled DNA will be fastest, linear moderate, and open circular will having the least velocity, just you can see in the diagram. So option D would be the correct answer. A protein sample when subjected to reducing SDS patch displayed two bands of molecular weight 20 and 25 kD. When the same protein sample was subjected to non-reducing isoelectric focusing, a single band was observed. These observations are best explained with the following relevant reasoning. The sample either contains two proteins of different molecular weight and same net charge, or one protein with two subunits that differ in molecular weight but share the same net charge. The sample contains two proteins of different molecular weight and same net charge, or the sample is heterodimer with the subunit sharing the same net charge but differing in the molecular weight. The experiment had some error and the results cannot be explained. Now, to understand this, let's see the figure. If we consider that a protein having two subunits, which can be connected with a disulfide linkages, and in the next case, there are two proteins with having a molecular weight of 20 and 25 kD. If you will run this protein on reducing SDS part, in the first case, if the disulfide bond will be reduced, then the 20 kD and 25 kD will give you two bands. While in the next case, since there is no disulfide bond is present in the protein, then it will also give you the same two bands. Since the molecular weight is not much different, so we can assume that the two subunits or the part of the single protein connected with the disulfide bond has the similar charges. So on isoelectric focusing, both the protein will give you single band. It cannot be differentiated whether the protein is connected with disulfide linkage and have two subunits, or there are two different proteins. If you really want to differentiate, then you should run a non-reducing SDS patch. If you will run a non-reducing SDS patch, then the first case will yield you a 45 kD band, while the second case yield you two bands of 20 kD and 25 kD. So here the option A seems to be close to correct. The sample either contains two proteins of different molecular weight and same net charge, or one protein with two subunits that differ in molecular weight but share the same net charge. The pi or isoelectric point of a protein is 6. When the electric field is applied at pH 8, the protein moves towards anode, moves toward cathode, does not move, or moves in random fashion. As we know that at isoelectric point, that is here, which is pH 6, the protein will be having equal number of positive and negative charge. That means it will be electrically neutral. When you keep any protein at very low pH, the protein develops the net positive charge. While if you keep any protein at an alkaline pH, they develop the net negative charge. So this protein whose pi is 6, if you keep it at a pH 8, then it is bound to develop a net negative charge. So definitely it will be attracted towards the positive electrode, that means anode. So the protein at pH 8 will be moving towards the anode. Isoelectric focusing is a technique routinely used to resolve protein based on their hydrophobicity, mass, pi, or length. Basically, during isoelectric focusing, the protein samples are resolved based on their pi, that is isoelectric pH. It is that pH, or isoelectric point is that pH basically at which the protein contains no net charge. That means they are electrically neutral. So option C is the correct answer. In counter immunoelectrophoresis, the antibody will migrate towards anode, the antibody will migrate towards cathode, electrophoresis will drive the antibody and antigen towards each other, or electrophoresis will drive the antibody and antigen parallel to each other. Countercurrent immunoelectrophoresis is a modification of immunoelectrophoresis in which antigen and antibody moves in the opposite direction and forms PPT in an area where they meet in concentration of optimal proportions. So option C would be the correct answer. Which of the following is a positively charged matrix for ion exchange chromatography? CM cellulose, phosphocellulose, nitrile triacetic acid, or DAE cellulose? DAE cellulose is a weak anion exchanger which may be used to bind negatively charged species. So DAE cellulose is having the positively charged matrix which can bind to the negatively charged species. So option D is the correct answer. The ligand used for affinity chromatography of a RNA containing poly A sequence is, obviously baat hai, oligo DT is the correct answer. Why? बिकॉज ओलाइगो डीटी जो है उसका वो कॉम्प्लीमेंट्री बेस पेयर है पॉली 18 का। तो अगर हम किसी भी मैट्रिक्स पे अगर ओलाइगो डीटी को टेथर कर दे तो ओलाइगो डीटी कॉलम जो है वो एक अफिनिटी क्रोमेटोग्राफी की तरह काम करेगा, विच विल बाइंड टू द एमआरएनए विच इज हैविंग पॉली 18 और उसको आप बाद में सेपरेट कर सकते हो। तो ऑप्शन सी शुड बी द करेक्ट आंसर। Lectins are known to bind to only ceramide, cholesterol, phospholipids or glycoconjugates. Lectins are the glycoproteins which was first discovered in plant and later in certain other organisms. They generally recognize and bind to specific sugar or something which is having sugar attached to them. So option D is the correct answer, that glycoconjugate which is having the molecule which is having attached sugar, they can be recognized by the lectin. Phase contrast microscopy, the rate at which light passes through the object is, चार ऑप्शन दे रखे हैं, inversely proportional to their refractive index, constant, directly proportional to their refractive index, exponentially related to their refractive index. Generally, in PCM of phase contrast microscopy, rate at which light travels through the object is inversely proportional to their refractive index. Generally, जो phase है light का, अगर if it is passing through an object of higher refractive index, then it's of surrounding, then इसका जो rate है वो retard कर जाता है। तो इसलिए option A should be the correct answer, that in phase contrast microscopy, the rate at which light passes through the object is inversely proportional to their refractive index. आगे चलते हैं। What is the effect of urea and formamide on DNA? You should remember that both urea and formamide, they decrease the TM of a DNA, means they promote DNA melting even at a low temperature. Hence, urea and formamide both denature the DNA, और इसीलिए option A should be the correct answer, that they decrease the TM of a DNA. Urea and formamide can decrease TM of the DNA, does not affect denaturation of the DNA, increase TM of the DNA, helps faster re-annealing of the DNA. Now, as we know that urea is an hydrogen bond donor as well as acceptor, so it may disturb the hydrogen bonding of nucleic acid. Formamide, on the other hand, it lowers the DNA stability by altering the ion solvating power and solubility of free bases. Now, both of these denaturants can denature our DNA and hence helps in reducing the TM. Always remember, whenever it is easy to denature the DNA, we require lower temperature to denature the DNA. So that is what TM is all about. So urea and formamide, being a denaturating agent, can help in denaturating the DNA at a lower temperature. So option A would be the correct answer. Specificity or stringency of a PCR reaction can be controlled by altering the reaction. और इसमें चार ऑप्शन दे रखे हैं आपको, वॉल्यूम, टारगेट सीक्वेंस, टेम्परेचर एंड सॉल्ट कंसंट्रेशन एंड टेम्पलेट कॉपीज। अगर हम ध्यान से देखें तो स्ट्रिंजेंसी ऑफ अ पीसीआर कैन बी कंट्रोल्ड by altering the temperature of DNA primer hybridization. As you know, as the temperature decreases, more mismatch can be tolerated. और अगर एक दूसरा फैक्ट अगर हम ध्यान से देखें कि प्रेजेंस ऑफ सॉल्ट, दैट्स आल्सो ग्रेटली इन्फ्लुएंस द पीसीआर एम्प्लीफिकेशन। इसके अलावा कुछ और फैक्टर्स भी हैं जो पीसीआर की स्ट्रिंजेंसी को कंट्रोल कर सकते हैं, जैसे कि प्राइमर्स की प्योरिटी, मैग्नीशियम आयंस, एडिटिव्स। बट इन दिस केस द ऑप्शन सी शुड बी द करेक्ट आंसर, दैट टेम्परेचर एंड सॉल्ट कंसंट्रेशन बोथ कैन strongly influence the specificity or stringency of a PCR reaction. Which of the following reagents are not a part of a typical polymerase chain reaction? Forward and reverse primer, definitely we require. DNA polymerase, here is the twist, we require thermostable DNA polymerase, not the any random DNA polymerase. Magnesium chloride, again we require, and nucleotide triphosphate, that's the twist. We require deoxynucleotide triphosphate for conducting a PCR rather than NTPs. So option D is the correct answer. What is approximately the half-life of P32? 30 days, 42 days, 14 days or 28 days? P32, it disintegrates by emitting a beta negative particle with a half-life of 14.263 days. That means the option C would be the close to correct. Which one of the following radioisotopes does not emit beta rays? Carbon, hydrogen, phosphorus or iodine? As we know that C14, tritium, phosphorus 32 They all are the beta emitter, while iodine 125 is an example of gamma emitter. So option D is the correct answer. The mammalian cells are routinely cultured under in vitro conditions at 37 degrees Celsius at 5% oxygen, 37 degrees Celsius and 5% carbon dioxide, 25 degrees Celsius and 5% oxygen or 5% carbon dioxide. Generally, mammalian cell cultures are maintained at a temperature of 37 degrees Celsius, 5% carbon dioxide and 95% of relative humidity. So option B is the correct answer. The arrangement of nucleotides in DNA can be seen by ultracentrifugation, X-ray crystallography, light microscope or electron microscope. Nucleotide arrangement in DNA can be seen by the X-ray crystallography, in which incident X-ray beams passes through the crystalline structure and gets diffracted into many specific directions. Watson and Crick proposed double helical structures of DNA based on the classical X-ray diffraction photo obtained by the Rosalind Franklin, which is also known as photo 51. So here the X-ray crystallography is the method of choice which can be used to see the arrangement of nucleotides in DNA. Which of the following technique is used to obtain the 3D structure of large proteins at atomic resolution? X-ray crystallography, NMR spectroscopy, electron microscopy or circular dichroscopy. X-ray crystallography has been used in past to resolve the 3D structure of biomolecules including proteins and DNA structure. Who can forget the contribution made by Kendrew in order to determine the first crystal structure of proteins of myoglobin? The underlying principle of using X-ray crystallography is that the crystalline atom causes a beam of X-ray diffraction into many specific directions. By measuring the angle and intensity of these diffraction patterns, a crystallographer can predict a 3D model of the electron density map within a crystal, which can later be used to determine the 3D structure of a protein. So, option A is the correct answer. Arrange the following landmark achievement in a chronological order: crystal structure of DNA, draft sequence of human genome, sequence of insulin, crystal structure of myoglobin. So we have to choose again the correct order in the chronology as they happen. If we know that insulin structure was first proposed by Frederick Sanger in 1951, for he was awarded Nobel Prize. In 1952, Rosalind Franklin took a detailed X-ray image of bDNA, using which Watson and Crick proposed the DNA structure in 1953. In 1957, John Cantu became the first person to resolve the crystal structure of the myoglobin, which was the first protein structure resolved by the X-ray crystallography. Human Genome Project was basically completed by 2001 to 2002. So the chronological order would be C, which was a sequence of insulin, A, then the crystal of DNA, crystal structure of the DNA, sequence of insulin 1951, crystal structure of DNA 1952, and followed by Watson Crick proposal 1953. Then next would be crystal structure of the myoglobin in 1957, and then draft sequence of human genome, that is around 2001. So option A is the correct answer. Which of the following scientists was responsible for the first database in bioinformatics? Max Perutz, Margaret Dayhoff, Linus Pauling or Dorothy Rothschild? Margaret Bailey Dayhoff was a pioneer in the field of bioinformatics. She is credited for the creation of protein and nucleic acid databases, and she published first her work in the name of Atlas of Protein Sequences and Structure. Eventually, her data became the first online database. So, option B is correct answer, that Margaret Bailey Dayhoff was the first American lady which was responsible for the creation of first database in the field of bioinformatics. Balls and sticks in small r: solid sphere and rods representing atoms and bonds respectively, cylinders or spiral ribbons to represent alpha helices and broad flat arrows to represent beta strands, line drawing representing bonds between the atoms, or solid spheres and rods representing bonds and atoms respectively. ResMol ball and stick model are the 3D models used to represent biomolecular structures, where each atoms are represented with a solid ball line spheres and stick represent bonds connecting two atoms. So here the ball and stick in ResMols are the solid spheres and rods representing atoms and the connecting bond respectively. चलिए वी आर हाफ वे थ्रू। इम्यूनोलॉजी में आपके दो टॉपिक बहुत इंपॉर्टेंट है, एंटीबॉडीज, उसके स्ट्रक्चर एंड जनरल प्रॉपर्टीज, एंड वैक्सीनेशंस। इन दोनों को ही आपको अच्छे से करके जाना होगा अगर आपको एमएससी बायोकेमिस्ट्री में रूटीन क्वेश्चंस को सॉल्व करना है। ठीक है, चलो देखते हैं। Humoral immune response in higher animals is involved with B cells, T cells, both B and T cells, or macrophages. See, it's a very confusing question. Whenever we hear humoral word, B cells is automatically comes in our mind. But remember, specific humoral activity actually involves the action of B cells or antibodies under the guidance of T helper cells. So answer has to be the both B and T cells. The class of immunoglobulin present in highest concentration in the blood of a human newborn is IgA, IgM, IgE, or IgG. The primary immunoglobulin in newborn is the maternal IgG, which is coming through the placenta. Remember, immunoglobulin G is the only antibody which can cross the placenta, which is coming from the maternal sides. So option D is the correct answer. Which immunoglobulin is predominantly in the primary immune response? IgG and IgM, IgM, IgE, or IgG? IgM is the predominant antibody during a primary challenge to an antigen, and upon encounter with antigen, it switched to either IgA, IgG, or even IgE depending on the type and the place at which the antigen was encountered, and it generally changes to either of these classes. So option B is the correct answer. There are various subclasses of antibodies found in the body fluids and body secretions. Many different functions may be attributed to these subclasses. Given below in list 1 is major function of different subclasses, and list 2 consists of the name of the subclasses. So there are various functions which are being given in list 1 item, while we have to correlate it the what is the major function played by the which antibody. So now list 1 first item would be binds to macrophages by Fc. Macrophages and monocytes possess a surface receptor which can bind to the Fc region of IgG and plays its role in phagocytosis. So A list item has to be connected with the 4 IgG. Now next is binds to mast cells by basophils. IgE binds to the mast cell in tissues and basophils in circulations and generally involved in the allergic reaction. So B component has to interact with the third, that is IgE. Now C is first B cell receptor. IgM is the first B cell receptor as well as the first antibody molecule present in newborns. So C has to be connected with fifth, that is IgM. Now D is no major specific function known other than antigen binding. Definitely it has to be IgD because IgD is not known to have a specific function, and it is debatable that why it is present in immune system. So D is connected with the 2. Now protection of mucous membrane. So IgA is an antibody which are associated with mucosal membrane. So E has to be connected with first. So if we see, then A is connected with fourth, B is connected with third, C is connected with fifth, D is connected with second, and E is connected with first, which is option D. IgM antibody can bind to 2 epitope, 10 epitope, 4 epitopes, or 1 epitopes. In its secreted form, IgM is a pentamer which is composed of 5 antibody molecules which can recognize and bind 10 epitopes. So option B is the correct answer. Which of the following is the main immunoglobulin in the gut and secretion like saliva, milk, tear, and is important in mucosal immunity? Is it IgA, IgM, IgD, or IgG? IgA is the main antibody which is found in the mucosal secretion like tear, saliva, respiratory or intestinal secretions, or even colostrum. Colostrum, which is the first milk produced by the lactating mother. So option A is the correct answer, that IgA is basically involved in the gut and secretion which is involved in the mucosal immunity. Immunoglobulin which takes part in hypersensitivity reaction. IgE is basically used in the type 1 hypersensitivity जो कि इन्वॉल्व करती है एलर्जिक, एटोपी और एनाफाइलेक्टिक शॉक्स. तो इसीलिए इसका ऑप्शन का आंसर होगा ऑप्शन बी, दैट इज आईजी. The immunoglobulin fold primarily comprises of gamma turn, alpha helices, beta sheets, or unstructured loop. Immunoglobulin fold consists of a pair of beta sheets, each built of antiparallel beta strand that surrounds a central hydrophobic core that is bridged by a single disulfide bond. So option C would be the correct answer, that beta sheets are responsible for making the immunoglobulin fold. Which of the following diseases are related to coronavirus? MERS, cholera, polio, or smallpox? You must be wondering in past 2-3 years that how come coronavirus has infected the humans which was earlier supposed to be found in the bats. Definitely coronavirus is a zoonotic virus which is found in the animals and has a potential to affect the humans when they comes in contact. But you might not be aware that in 2012, a similar kind of a virus, which is also known as MERS virus, has been found in the Gulf countries, which is supposed to be infected the camel first and then from camels has been transmitted to the humans. So MERS is also a type of coronavirus which has already affected the humans in 2012. B cells acquire immune competence in thymus, bone marrow, liver, or spleen. Development of a B cell begins in the bone marrow and in sequential manner they transform from pro B cell to immature B cells. Now immature B cells once formed, they are having expression of BCR, which is a cell surface receptor. The cell surface receptor is once surfaced, then we can say that B cells becomes immunocompetent. So B cell actually acquire immune competence in bone marrow. Antigen presenting cells that activate helper T cells must express which one of the following on their surfaces? Ig, class 1 MSc antigens, class 2 MSc antigens, or gamma interferon. T helper cells or CD4 positive helper cells, they recognize and interact with the antigens which are bound to the class 2 MSc molecule present over the membrane of antigen presenting cells. They also require an additionally co-stimulatory signals which is produced by the APCs, which led to the activation of T helper cells. So basically class 2 MSc antigen bound receptor which are present over the APC, and also a co-stimulatory signal is required for the activation of helper T cells by the APCs. Surface marker of helper T cells are CD8, CD4, CD48, or CD38. CD4 is a characteristic CD or cluster of differentiation for TH cell, while CD8 is a marker of cytotoxic T cells. While all the T cells, whatsoever it is, whether it's a helper T cells or cytotoxic T cells or Treg cells, they all contain CD3 as a marker of all T cells. So here, the all T cell lineage will have CD3. Specific marker for helper T cells is CD4. Specific marker of cytotoxic T cells is CD8. HIV infected patients exhibit preferential decrease in CD4 cell count, CD8 cell count, CD20 cell count, or CD40 cell count. HIV invade T helper immune cells, which is also known as CD4 positive T cells, and once upon infection, it causes the decline of T helper cells. If it falls below a critical level, then our body becomes susceptible towards the opportunistic infection. So basically, HIV infect patients exhibit preferential decline in CD4 or T helper cell counts. Identify the correct name of a protein present only in the biological membrane. Influenza, hemagglutinin, BSA, insulin, or trypsin. Influenza hemagglutinin HA is a homotrimeric membrane glycoprotein found on the surface of the influenza flu virus, and it is responsible for its infection along with an enzyme called neuraminidase. So here the option A has to be the correct answer. The classical complement pathway is initiated by interaction of C1 with antigen IgG complex, antigen, factor B, or bacterial lipopolysaccharide. The classical pathway of complement system is initiated by IgM or IgG dependent antigen antibody complex, which bind to the C1q, which is the first protein of the cascade. Following activation, a series of proteins are recruited to generate the C3 convertase. The C3 convertase cleave the C3 protein. So the classical complement pathway is initiated by the interaction of C1 with antigen IgG complex. Kupffer cells are a specialized type of a lymphocyte, macrophages, dendritic cells, or monocyte. That's very simple. Kupffer cell, these are the specialized macrophages which are found in the sinusoid of the liver, where they are phagocytic in nature. So option B is the correct answer. Receptors present on macrophages can bind to. We have just discussed that macrophage possesses receptor for the Fc region of the IgG, which is option A. Grafts between genetically identical individuals, that is identical twins, are not rejected if a kidney is grafted, but skin grafts are rejected, are not rejected even without immunosuppression, are subjected to hyperacute rejection, or rejected slowly as a result of minor histocompatibility antigens. Basically, what happens is, barring technical difficulties, a graft between genetically identical twins or individuals is readily accepted and are almost never rejected. Such grafts are also known as isograft, as you can see it in the figure. So now here the option B is the correct answer, that grafts between genetically identical individuals or identical twins are not rejected even without using immunosuppression. Hybridomer technology uses splenocytes for the production of, that's very easy, that hybridomer technology is used for the production of monoclonal antibody, where these antibodies specific to the antigen of interest, which is not an autoimmune disorder: grave disease, Alzheimer, multiple sclerosis, or celiac disease. A syndrome in which our immune system wrongly targets our own body is called as autoimmune disorders. Some of the common examples of autoimmune diseases are rheumatoid arthritis, Hashimoto thyroiditis, celiac disease, grave disease, rheumatic fever, multiple sclerosis, or pernicious anemia. Alzheimer's disease, on the other hand, it's a progressive neurodegenerative disorder which is caused due to the death of brain cells over a period of time. So generally a person acquire in their own life the Alzheimer's disease. So option B is the correct answer. सेल बायोलॉजी के जो क्वेश्चंस होते हैं वो बड़े स्पेसिफिक क्वेश्चंस होते हैं. मेम्ब्रेन से एक इंपॉर्टेंट टॉपिक है, बहुत क्वेश्चंस आए हुए हैं पास्ट ईयर में. और सेल डिवीजन अच्छे से कर लीजिएगा. सेल डिवीजन एंड मेम्ब्रेन दोनों में से आप क्वेश्चन एक्सपेक्ट करिए इस साल भी. ओके. Which one is the heaviest particulate component of the cell? Nucleus, mitochondria, cytoplasm, or Golgi apparatus? Basically, we have to find out which is the most dense organelle present in the cell. So, as we know that nucleus is the heaviest particulate component of the cell, having diameter of around 4 to 6 micrometer. So, option A is the correct answer. Which of the following is a non-membranous structure? Golgi body, microbodies, inclusion bodies, or glyoxysomes. Inclusion bodies are the tiny particles that freely suspended and floating within a cytoplasmic matrix. while rest are the example of membranous organelles. So option C is the correct answer. Absence of functioning peroxisomes results in Woolman disease, Zellweger syndrome, Hunter syndrome, or Gaucher disease. Zellweger syndrome is the most common peroxisomal genetic disorder which happens due to the absence of functional peroxisome. So option B is the correct answer. Several classes of hydrolases are localized in. Again, one of the most simple question that in the lysosomes, there are various types of hydrolases are present to degrade various biomolecules category, including proteases, lipases, nucleases, or even glucosidases. Cell-to-cell fusion is exhibited by the formation of multinucleated cells, microvilli, elevated numbers of endosomes, or numerous vacuoles. Cell fusion is an important cellular process in which several uninucleated cells, that means cells with a single nucleus, may combine to form the multinucleated cells, which is also called as syncytium. So option A is the correct answer. All of the following statements are true about lysozyme except which one? It hydrolyzes peptidoglycan, it destroys gram-positive cell walls, it is an organelle in WBCs, or it is an enzyme. Lysozyme is an enzyme which is often present in the body secretion like tears, saliva, or even milk. It hydrolyzes the beta-1,4 glycosidic bond between NAG and NAM of the peptidoglycan layer. Peptidoglycan layer is comparatively more in the gram-positive than gram-negative bacteria, and hence they are very effective against the gram-positive bacteria. As far as third option is concerned, that it is an organelle in WBC, is the incorrect statement about the lysozyme. So option C is the correct answer. The acid hydrolases of lysosomes do not digest the organelle itself because lysosomal milieu does not provide optimum pH. These enzymes are present as zymogens. Lysosomal membrane is heavily glycosylated, or ultimately the lysosomes are also degraded. Lysosomal enzymes cannot digest themselves because most of the membrane protein of lysosomal membranes are heavily glycosylated. So option C has to be the correct answer. Acid phosphatase can be used as a marker enzyme for lysosomes, peroxisomes, nucleus, or mitochondria. Marker enzymes are those enzymes which tend to remain confined to a specific organelle or a tissue. Generally, the acid phosphatase is considered to be a marker enzyme for lysosomes, along with glucuronaminidase or N-acetyl D-hexosaminidase. So here the option A would be the correct answer. Given below are the events in the cell cycle. So there are 4 events which are being given, and we have to arrange as per the correct order in the occurrence. Option A was phosphorylation of lamin A, B, and C. B. Phosphorylation of retinoblastoma protein. Polyubiquitination of securin. Or D. Association of inner nuclear membrane protein and nuclear pore complex proteins with chromosomes. So we have to find out what is the correct order in a typical mammalian cell cycle. As we know that cell cycle event is tightly regulated by the cyclin and cyclin dependent kinases, which are definitely under controls. Retinoblastoma protein is a nuclear protein that undergoes in phosphorylation and dephosphorylation in concert with cell cycle. To begin cell cycle, retinoblastoma is phosphorylated, which now cannot bind to the E2F transcription factor. This transcription factor is required to begin the cell cycle. So when the decision has to be made to begin the cell cycle, E2F must be free. So retinoblastoma phosphorylation actually makes sure that E2F remain free for the beginning of cell cycle. That is the first step. Once cell cycle is decided to begin, then lamin has to be depolymerized. These architectural proteins remain associated with animal nuclear membrane, and phosphorylation of lamin-based depolymerization on the nuclear envelope break down before mitosis get complete. So that has to be the second step. One of the most important steps during mitosis is the separation of sister chromatid to the opposite poles. In order to do so, cohesin, a protein which glue the sister chromatids, need to be destroyed. And this destruction is being done by an enzyme called separase. Separase generally remain inactive due to the another protein called securin. So basically securin need to be destroyed in order to active the separase, which will ultimately going to deglue the cohesin. Polyubiquitination degrades securin, and in chain of events ultimately cohesin is degraded and finally anaphase is complete. So the polyubiquitination of securin should be the third step, which is occurring somewhere nearby anaphase. Finally, inner nuclear membrane complex and nuclear pore complex proteins regain its original position with chromosomes and a fresh nucleus is formed in a daughter cell. So the D steps has to be the last. So what should be the correct order? That the B, phosphorylation of retinoblastoma cells, retinoblastoma protein, which will make sure that it will free the E2F, so the cycle can begin. Second, the phosphorylation of lamin ABC, so that the nuclear membrane can be dissolved. The C, polyubiquitination of securin, which will make sure that the cohesin can be dissolved and sister chromatid can separate to the opposite pole. And last, that the new nuclear membrane will be formed. So the correct order would be B, A, C, D, which is option D. So option D has to be the correct answer. All the membrane proteins are known as G proteins because they have a subunit that activates adenylate cyclase, hydrolyze GTP, are gated ion channel, or mediate the effects of insulin on cells. That's very easy. All the G protein known today contains a canonical GTPase fold which can bind and hydrolyze GTP. So option B is the correct answer. The most abundant phospholipid in bacterial cell membrane is phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, or cholesterol. Bacterial plasma membrane are often composed of phosphatidylethanolamine, that is also known as neutral phospholipid, and contains no cholesterol. In general, gram-positive bacteria have higher content of phosphatidylethanolamine than gram-negative bacteria. So here the option C is the correct answer. Cellular membranes are self-sealing in nature. If they are punctured or disrupted mechanically, they quickly and automatically reseal. What properties of such sealing are responsible for this feature? Is it due to the hydrophobic effect of membrane lipids? Is it due to the hydrophilic effects of membrane lipids? Because of the charge to charge interaction among lipids, or due to the protein lipid interactions? See, in case of cellular membrane is punctured or broken, then hydrophobic fatty acid chains of the plasma membrane, they tries to aggregate, and it is happening because of the strong hydrophobic interaction which is present in the aqueous medium. Remember, the hydrophobic interaction seems to be maximum when they are placed in the watery medium or aqueous medium. Hence, when due to the puncture, these hydrophobic groups are separated in aqueous environment, they prefer to reseal. So, option A is the correct answer. Cellular membranes are self-sealing in nature. If they are punctured or disrupted mechanically, they quickly and automatically reseal. ये जो properties आती है ये hydrophobic interactions की वजह से आती है, because hydrophobic interaction they drive the membrane formation, और अगर कहीं पे भी उसे पंक्चर कर दिया जाए तो they have a property to reseal them. तो इसीलिए इसमें क्वेश्चन का आंसर होगा ऑप्शन ए, हाइड्रोफोबिक इफेक्ट्स ऑफ मेम्ब्रेन लिपिड्स. आप देख रहे हैं क्वेश्चन बहुत आसान है, बहुत सिंपल बेसिक कॉन्सेप्ट्स पे हैं. आगे चलते हैं. The observation that plasma membrane protein mix after cell fusion provide evidence for rotational movement of plasma membrane proteins, the bilayer structure of the biomolecules, the fluidic mosaic model, or interaction of plasma membrane proteins of two different cell types. An experiment performed by the Fry and Adidin provided evidence in support of the membrane fluidity and dynamism. First, you need to understand what was the previous model. Davidson and Daniele proposed a tri-layer model of the lipid bimembrane, which suggests that the lipid membrane used to be sandwiched between two protein layers. Now, when two cells will fuse, as per their model, the protein layer must get locked between two lipid bimembrane layers. But as far as Fry and Adidin reports were concerned, they found that the protein of both the layers, they get diffused and mingle, means that the layer are fluidic in nature and can mix. So option C is the correct answer, that the Fry and Adidin model suggested that the lipid bilayer are fluidic in nature rather than static. An integral membrane protein spanning lipid bilayer of the membrane must have only no hydrophobic domains, two hydrophobic domains, three hydrophobic domains, or one hydrophobic domains. A typical integral protein generally remain attached permanently to the biological membrane, and these intrinsic protein or integral protein must have at least one or more than that segments that are embedded in phospholipid layer and having the hydrophobic side chain residues. These hydrophobic side chain residues interact with the fatty acyl group of the membrane phospholipid. So here the option D is the correct answer, that a typical intrinsic or integral membrane protein must have at least one hydrophobic domains. Lipid bilayer membrane model was proposed by Daniel and Davidson, Singer, Henderson and Nigel Unwin, and Robertson. Daniel and Davidson, they proposed the sandwich model of lipid bilayer, in which a lipid bilayer was coated on either side by the hydrated protein. This model was further refined in 1970 by Singer and Nicholson, and they proposed a fluid mosaic model which is even accepted today. So here the option A is the correct answer, that the lipid bilayer originally was discovered or proposed by the Daniel and Davidson. Molecules that pass through a cell membrane most easily are small and hydrophobic, large and polar, ions, or monosaccharides. Small molecules that are hydrophobic or non-polar, that means have no charge, can cross the membrane easily through the cell membrane, while ions, like charged ones, and larger hydrophilic molecules cannot pass through. So here the option A has to be the correct answer, that small and hydrophobic molecules that pass through a cell membrane most easily. Sialic acid is a part of phosphatidylcholine, ganglioside, cholesterol, or triglycerides. Sialic acid are carbohydrate molecules with a C9 carbon backbone and a carboxylic acid group, which is commonly present as a part of glycoprotein, glycolipids, and gangliosides. So option B is the correct answer. Which of the following is not an ionophore? So before we solve this question, we should know what is ionophore. So ionophores are a class of compound that forms a complex with specific ion and facilitate their transport across the cell membrane. So now there are four options are given, like monensin, valinomycin, kanamycin, or gramicidin. So we should know which one is not an example of ionophore. The common example of ionophores are valinomycin, gramicidin, nystatin, monensin, while kanamycin is not an example of ionophores. Rather, kanamycin is used as an antibiotic which interfere the protein synthesis. It binds to the 30S subunits of bacterial ribosomes, which result in incorrect alignment of ribosomes over the mRNA, and hence a wrong amino acid is placed in a growing peptide. So option C is the correct answer, that kanamycin is not an ionophore. Which one is a true membrane protein? Glycophorin, isocitrate lyase, pyruvate kinase, or hemoglobin? As you can see in the figure, glycophorin A is a type 1 single span membrane protein of RBC with an extracellular N-terminal domain which is modified with sialic acid. Due to the sialic acid, it has a negative charge over the outer cell surface side. So option A is the correct answer, that glycophorin is a membrane bound protein. Multi-drug resistance of cancer cells are because of the appearance of high rate of protein secretion, increase of endocytotic capacity, elevated number of GM1 on cell surface, or special transporter protein on the plasma membrane. Multi-drug resistance or MDR in the cancer chemotherapy has been pointed out as the ability of cancer cells to survive against a wide range of anti-cancerous drugs. One of the mechanisms of MDR is enhanced efflux of drugs through the pre-specialized transporters like ABC transporters, which flush out the drug outside the cancer cell, leading to the less absorption of drug to these cells. So basically होगा क्या? The cancerous cells has got the ability now so that any anti-cancerous drugs which enters into it, they can be effluxed out, they can be thrown out, and hence they can sustain against these anti-cancerous drugs. So option D would be the correct answer. Which protein strictly interacts with phosphatidylcholine? Phosphorylase, phosphatase, phospholipase, or alkaline phosphatase? Phospholipase D is an enzyme which acts on phosphatidylcholine and hydrolyze to produce the signal molecule like phosphatidic acid and soluble choline. So option C is the correct answer. Receptor molecules on the cell surface for cholera toxin is glycophorin, EGFR, cholesterol, or gangliosides. The membrane receptor for cholera toxin on the cell membrane appears to be a complex which contains the ganglioside GM1. So option D is the correct answer. Mode of action of penicillin on killing bacterial cell is through its action on ribosome, plasma membrane, cell wall, or DNA. Penicillin kills bacteria through binding of beta-lactam ring to D-transpeptidase, which inhibits its cross-linking activity and preventing new cell wall formation. Definitely, without cell wall, a bacteria is vulnerable to osmotic pressure. So, option C is the correct answer. मॉलिक्यूलर बायोलॉजी के जो क्वेश्चंस होते हैं किसी भी दूसरे एग्जाम्स की तरह रेप्लिकेशन, ट्रांसक्रिप्शन, ट्रांसलेशन, जीन रेगुलेशंस, ऑपरॉन्स, ये जनरली द पेट क्वेश्चंस इनवाइट करते हैं, ठीक है ना, इंपॉर्टेंट क्वेश्चंस बनाते हैं. बट इंटरेस्टिंगली एमएससी बायोकेमिस्ट्री एग्जाम में हमने देखा पास्ट ईयर के क्वेश्चंस के दौरान कि यहाँ पर जो साइंटिस्ट के नाम एंड डिस्कवरर्स जो इंपॉर्टेंट फेज में आए हैं मॉलिक्यूलर बायोलॉजी डोमेन में, उन पे बहुत क्वेश्चंस आए हैं. तो कई साइंटिस्ट जो कि बड़े इंपॉर्टेंट फिगर रहे हैं मॉलिक्यूलर बायोलॉजी में, उनका आप एक बार गो थ्रू जरूर कर लीजिएगा, ठीक है. बाकी के रूटीन क्वेश्चंस ही आएंगे, देखेंगे भी अभी हम. The enzyme responsible for conversion of negatively supercoiled DNA into a relaxed circular DNA is DNA gyrase, DNA topoisomerase 1, DNA topo 2, or DNA ligase. Generally, topoisomerase 1 introduces a single-stranded break in duplex DNA and remove supercoiling to the relaxed state. Now, topo 2, which is also known as DNA gyrase, is responsible for converting bacterial circular DNA to the negatively supercoiled DNA. That means what? Topo 1 actually releases negative supercoiling to make a bacterial circular DNA as a relaxed DNA. While topo 2, or also known as DNA gyrase, it actually introduces negative supercoiling. So here the option B has to be the correct answer. The scientists who identified the transforming agents in Griffith's famous experiment as DNA was Irving Chauger, Oswald Avery, Frederick Michel, or Peter Michel. In 1944, Oswald Avery, MacLeod McCarty, and Colin MacLeod, they identified the transforming agent in Griffith experiment was actually DNA. So here the option B has to be the correct answer. Match the names of the scientists in list 1 with their achievement in list 2 and choose the correct answers given below. So list 1 outlines the name of the scientists and we have to correctly match with their discoveries. First one is Watson and Crick. In 1953, Watson and Crick proposed the 3D double helical structures of DNA. So A has to go with the third. Now Robert W. Hawley, he won the Nobel Prize in Medicine in 1968 for describing the structure of tRNA for alanine, and thereby he linked the DNA with the protein synthesis. Actually, he shared Nobel Prize with Hargobind Khurana and Marshall Niederberg, who contributed for the cracking the genetic code mystery. So B has to go with the fourth, that Robert W. Hawley has actually proposed the Clover model of tRNA, while Marshall Niederberg, they are basically involved with the deciphering the genetic codon. Now, Monod and Jacob are famous for their work on E. coli lacopron, while John Jeffrey is better known for developing techniques for genetic fingerprinting and DNA profiling. So D has to go with the fifth and E has to go with the first. So if we find out, then option B is the correct answer. Arrange the following events of meiosis in correct sequence: crossing over, synapsis, terminalization of chiasmata, or disappearance of nucleolus. Steps will be chromatin condensation, which happens to be in leptotin; synapsis, which happens to be in the zygote; crossing over, which happens to be in the pechite; chiasmata appearance, which has to be in the diplote, while chiasmata terminalization followed by nucleus disappearance, that has to be in the dikinesis. So the option has to be B, synapsis first; A, crossing over second; C, terminalization of chiasmata third; D, disappearance of the nucleolus, that is D. So option BACD is the correct sequence, that is option B. 5' end capping of mRNA transcripts in eukaryotes involve the following events: A, addition of GMP on the 5' end; B, removal of gamma phosphate of the triphosphate on the first base at the 5' end; 5 to 5' linkage between GMP and the first base at the 5' end; or D, addition of the methyl group to N7 position of guanine. Now we have to arrange in a correct sequence. If you see it in the figure, that the steps of 5' cappings are: first, gamma phosphate removal at the 5' end; second, the addition of GMP at the 5' end; third, condensation of GMP with beta phosphate at the 5' end. Now last, the addition of methyl group at the N7 of guanine. If you correctly sequentially arrange these, then the B steps come to the first, A steps come second, C steps come third, and D steps come last. So B, A, C, D has to be the correct option, that means option C. Arrange the following steps of protein synthesis in the order in which they occur within a cell. DNA unzip in the nucleus, tRNA brings in proper amino acid from the cytoplasm, mRNA attaches to the ribosomes and first codon is read, mRNA nucleotide transcribes the complementary DNA message, mRNA leaves the nucleus and goes to the ribosome, and F, a second tRNA brings in the new amino acid. So we have to choose the correct answer from the options given below as per their sequence. So if you see it in the diagram, what is the first step is going to happen? DNA is going to unzip for the transcription process. What is the next process? Then the mRNA nucleotide transcribes the complementary DNA message, which is being done by the RNA polymerase, which uses the DNA template and synthesizes the mRNA. Now what is the next step? mRNA is going to leave the nucleus and going to cytoplasm and look for the ribosomes. Now what is the next step? Now this mRNA is going to attach to the ribosomes and first codon is read. What is the next step? tRNA brings in the proper amino acid from the cytoplasm, which is generally used to be the first codon is always AUG, and generally the methionine is being attached at the N terminal. What is the last step? A second tRNA brings in the new amino acid as per the second codons. So here if you see, if you arrange in a systematic way, then A will be followed by D, will be followed by E, will be followed by C, then B, and then last would be the F, which is an option C. During DNA replication, which of the following enzyme is involved in removing supercoiling ahead of the replication fork? That's very easy. Primase, polymerase, helicase, or topoisomerase. Basically, topoisomerase helps in relaxing supercoiled DNA to allow the initiation of a smooth replication event. So option D is the correct answer. A regulatory control sites of a gene must be very close to the gene it affects, must lie upstream of the gene it affects, is transacting or is cisacting. Regulatory genes is generally involved in controlling the expression of one or more genes. Such sequence can be located within, adjacent, or far away from the genes which are regulated. More often, these regulatory sequences are present upstream or 5' end to the start of the regulated gene. So, option B would be the close to correct. RNA-dependent DNA polymerases are known as transcriptases, primases, recombinases, or reverse transcriptases. So any DNA polymerases which are using RNA as a template in order to synthesize the DNA, they are called as reverse transcriptases. So reverse transcriptases are RNA-dependent DNA polymerases that make the cDNA from RNA template. This kind of enzymes was first discovered by the Baltimore in HIV, and for which he was awarded Nobel Prize. Rifampicin is an inhibitor of DNA replication, DNA gyrase, translation, or transcription. Rifampicin is a known inhibitor of RNA polymerase. When upon binding, it actually halts the transcription process. Interestingly, this antibiotic actually inhibits the prokaryotes because it's a specific inhibitor of prokaryotic RNA polymerase and do not influence the eukaryotic RNA polymerase. So option D is the correct answer. Which of the following molecules can be utilized for establishing early evolutionary processes? Ribosomal RNA, mitochondrial DNA, chloroplast, or nuclear DNA? 16S ribosomal RNA gene is a universal marker for phylogenetic reconstruction because of its high conservation. So, ribosomal RNA is a established marker for establishing the early evolutionary processes. Professor Howard Temin is famous for restriction endonucleases, DNA polymerases, DNA ligases, or reverse transcriptases. Professor Howard Temin was a cancer researcher who won the Nobel Prize for his role in discovering reverse transcriptase along with Baltimore. Restriction endonuclease was discovered by Auber, DNA polymerase was discovered by Kornberg, while DNA ligase was discovered by the Martigallard. So option D is the correct answer. Professor Hargobind Khurana is known to first synthesize polynucleotide, amino acid, protein, or siRNA. In 1968, the Nobel Prize in Physiology or Medicine was jointly awarded to Robert W. Hawley, Hargobind Khurana, and Marshall W. Nirenberg for their interpretation of the genetic codon and its function in the protein synthesis. Khurana was one of the first scientists to demonstrate the role of nucleotide in protein synthesis and helped in cracking the genetic codon. So he is basically first known to synthesize the polynucleotide. So option A is the correct answer. Which of the following amino acid is coded by a single codon? Phenylalanine, arginine, tryptophan, or lysine? Tryptophan and methionine are the only two known amino acids which are being coded by only single codon. Like tryptophan is coded by UGG while methionine is coded by AUG, which is also a first codon. Rest 18 amino acids are encoded by at least 2 and maximum of 6 codons. So option C would be the correct answer. Very last step of a protein secretion from live cell is somoylation, endocytosis, membrane fusion, or protein sorting. A conventional secretory pathway of a secretory protein, it starts with an N-terminal ladder peptide which derive the nascent protein to the ER. Now these proteins are transported to cis-Golgi network from rough endoplasmic reticulum through vesicle. Here the certain post-translational modification takes place, after which these proteins are again packed in the vesicle, which goes to the membrane, fuse, and releases the proteins out of the cell. So here the option C would be the last stage of a protein secretion from a live cell. Signal hypothesis for protein trafficking was proposed by, इसमें आंसर होगा गुंटर ब्लोबल, because Gunter Blobel and David Sabatini in 1971 they proposed the signal hypothesis. उसके हिसाब से जो भी प्रोटीन जो साइटोसोल में पड़े हैं वो कैसे किसी ऑर्गेनल में डायरेक्ट होते हैं, जैसे ईआर और ईआर मेम्ब्रेन में, तो उसके लिए एज पर देम जनरली इन प्रोटीन्स में एक सिग्नलिंग पेप्टाइड होता है which actually helps them, these proteins, to direct or traffic to a particular organelle, और इसीलिए इसका ऑप्शन आंसर होगा डी, that is Gunter Blobel. This is the picture of Gunter Blobel, and option D should be the correct answer. The scientist who created the first recombinant DNA molecule was Howard Temin, Paul Werk, Siemens Shapiro, or Ian Wilmot. In 1972, Paul Werk made the first recombinant DNA molecule using ligase, while in the same year, Stanley Cohen, Annie Cheng, and Herbert Boyer produced the first recombinant DNA organism. So, option B has to be the correct answer. Protein is degraded inside living cells through a key process of farnesylation, ubiquitination, methylation, or palmitoylation. Ubiquitination is a process in which a protein subjected to degradation is tethered with a ubiquitinin sequence and recognized and degraded by the proteasome. So, option B is the correct answer. Only one X chromosome in the aneuploidy females is a characteristic of individual with Cry-to-Cat syndrome, Klinefelter syndrome, Down syndrome, or Turner syndrome. Sex chromosome aneuploids comprises loss or gain of sex chromosome, that is X chromosome. So generally, all aneuploid females, they generally have either a higher copy of the sex chromosome, X chromosome, or there is a loss of a sex chromosome, that means X chromosome. Turner syndrome is a loss of X chromosome, while Klinefelter is a gain of X syndrome. So in the question we have been asked that only one X chromosome is there. That means out of a two X chromosome, one is lost, which is the characteristic of a Turner syndrome. During UV radiation of DNA, the following compound is formed. That's very easy. Thymine dimer is a photolesin which is produced due to the exposure of UV radiation to a DNA. Hence, option A has to be the correct answer. AP endonuclease is responsible for double strand break repair, base excision repair, nucleotide excision repair, or mismatch repair. AP endonuclease is an enzyme that is involved in the DNA base excision repair pathway. Its main role is to introduce a nick in the DNA next to an AP site. AP sites is a place where there is a purine or pyrimidine base is missing. So option B is the correct answer. Which of the following would be the enzyme of choice to carry out the nick translation reaction? E. coli DNA pol 1, T4 DNA polymerase, T7 DNA polymerase, or Linau fragment. Nick translation is used to label the hybridization probe, which can be used further in blotting like northern or southern, or even microarrays. Basically, there are two enzymes which are important in nick translation. DNase 1, which is used to introduce the nick in DNA duplex, while other enzyme is DNA pol 1, also known as Kornberg enzyme. It has a two unique activity. One, 5' to 3' pol activity, through which it can introduce the new fresh nucleotide at the 3' end, while another activity, which is a 5' to 3' exonuclease activity, which can be used to digest the non-template strand. So here the Kornberg enzyme or E. coli DNA pol 1 is very much essential in nick translation event. So option A is the correct answer. Which enzyme carries the incision of 3' position of the lysine in nucleotide excision repair? URBB, URBC, URBD, or URBA? Nucleotide excision repair is a kind of a DNA repair mechanism against damages which is caused by chemicals like intercalator, radiation, or even mutagens. URBABC is a multi-enzyme complex which is associated with DNA repair, and it removes the 12 nucleotide patch from the mutation site, which is later replaced by the DNA polymerase by adding the correct nucleotides. Let's understand the steps. What happens is, once the radiation causes the pyrimidine dimer formation like thymine dimer, it is being recognized by the URBA and URBB dimer complex. Once recognized, then what happens is the DNA, it wraps around the URBB. Once it is being done, then URBA left the place. As the URBA goes off, then URBC comes, which is an endonuclease, and it recognizes the site and cuts the 8 base pair upstream of the mutation site and 4 base pair downstream of the mutation site. That means a whole sum of 12 nucleotide strand. Once it is being cleaved off, then a helicases come, which is URBD, and which opens up the DNA. Now this DNA which is opened up is being filled up by the polymerases, and finally ligase ligate the two strands. So endonuclease URBC actually causes the incision of 3' position of the lesion in nucleotide excision repair. Muscular dystrophy is a result of mutation in glycophorin A, mutation in N-chirin, mutation in BAN3 protein, or mutation in dystrophin. Mutation in the DMD gene causes the Dechne and Baker form of a muscular dystrophy. DMD gene provides instruction for making a 4 to 7 kD cytoskeleton dystrophin protein, which is required to stabilize and protecting the muscle fiber. So hence option D is the correct answer. सरप्राइजिंगली ड्यू एमएससी बायोकेमिस्ट्री के एग्जाम्स में हमने देखा जेनेटिक्स में बहुत क्वेश्चंस पूछे नहीं गए हैं. जो क्लासिकल क्वेश्चंस आते हैं जेनेटिक्स के, एटलीस्ट वी हैव नॉट दैट मच एनकाउंटर्ड, राइट. हो सकता है इस साल आ जाए, बट जेनेटिक्स के आप बेसिक्स क्वेश्चन जरूर करिएगा, स्पेशली जो बैक्टीरियल में हॉरिजॉन्टल जीन ट्रांसफर है उसको अच्छे से कर लीजिएगा. हो सकता है वो एक क्वेश्चन बन जाए. ओके. Scientist who proved that DNA and not the protein as a genetic material, was it Posture, Jenner, Hershey and Chase, or Beadle and Tatum? Hershey and Chase experiment were a series of experiments which helped to confirm that DNA is a genetic material. So basically उन्होंने क्या किया था? They have labeled the DNA with phosphorus 32. Remember, phosphorus is only present as a part of DNA, not as a part of a protein. While they labeled the protein code with the sulfur, which is part of only amino acid category, not the DNA. तो DNA भी label हो गया specifically, and protein code was also labeled. Now they infected this bacteriophage with the bacteria. Abhi now they have traced the radioactivity, where it is going. Since the radioactivity was remained in the bacteria, that simply means that bacteria must have got the radioactivity because of the viruses, and this radioactivity was rich in P32. That means bacteriophage must have introduced their DNA as a genetic material rather than protein. So these experiments confirm that the DNA is the genetic material, not the protein. Option C is the correct answer. Bacterial conjugation was discovered by Lederberg and Tatum, Beadle and Tatum, Griffith, or Robert Koch. Conjugation, it was first discovered in 1946 by Edward Tatum and Joshua Lederberg when they were studying the two strains of E. coli with different nutritional requirement. There they found that the certain gene was passed on from one bacterial species to the another bacterial species. So this was the first discovery of bacterial conjugation. Which of the following can be used to construct a linkage map of the HFR chromosome? HFR cells जो होती हैं, they have a high frequency of recombination between F factor plasmid and main chromosomes, or generally it happens at a certain similar insertion sequences. Each gene enters the F negative cells at a characteristic time, and hence a linkage map can be constructed using a time of entry as a measure of for HFR chromosome. तो इसीलिए इसका आंसर होगा ऑप्शन बी, दैट इज टाइम ऑफ एंट्री. अब तक आपने बहुत सारे क्वेश्चंस को कर लिया है, अच्छा खासा पूल को आप कवर कर रहे हैं, और ये सारे ही क्वेश्चन पास्ट ईयर में आए हुए हैं. एनिमल फिजियो में भी आपके जो है क्वेश्चंस इनवाइट होते हैं, डाइजेशन से बहुत क्वेश्चंस आए हुए हैं, एंड साथ में हार्मोन्स पे क्वेश्चंस हैं. ठीक है, तो उनको भी आप एक बार गो थ्रू कर लीजिएगा, और इसके बाद हम लास्ट डोमेन करेंगे प्लांट बायोकेमिस्ट्री. Human intestinal flora cannot digest cellulose, lignin, starch, or pectin. Human body does not have the digestive mechanism to break the monosaccharide bond of a cellulose, so we cannot digest the cellulose. What are bile salts? Charged phospholipid, amphipathic cholesterol analog with detergent properties, esterified cholesterol, or hydrolyzed form of a triglycerol. Naturally occurring bile salts are derived from the cholesterol and are amphipathic detergent types molecule. In water, they form micelles which are required for fat emulsification. So option B has to be the correct answer. Given below are the two statements. One is labeled as assertion and other is labeled as reason. Assertion is bile juice helps in the digestion of food in small intestine. Reason is that bile juice contains sodium salts. So in the light of our statements, choose the correct option from the options given below. Both A and R are true and R is the correct explanation of A. Both A and R are true but R is not the correct explanation of A. A is true but R is false. A is false but R is true. As we know that the bile salts helps in the lipid digestion in the small intestine. Bile juice has the bile salts which actually break down the large fat globules into small particles, which is also known as emulsification of fat. Now, due to the emulsification of the fat, pancreatic lipase can act over it. And the main component of a bile juice are the bile salts which are composed of the sodium salts of glycine and taurine. So both assertion is correct that bile juice helps in the digestion of food, especially fat in the small intestine, and reason is also correct that bile juice contains sodium salts of glycine and taurine. So option A is the correct answer. The role of cholecystokinin is to stimulate gastric motility, stimulate enzyme secretion from pancreas, inhibit enzyme secretion from pancreas, or inhibit gastric motility. CCK or cholecystokinin, it stimulates pancreatic acinar cells to release a juice rich in pancreatic digestive enzymes, which catalyze the fat, carbohydrate, and protein digestions. So option B is the correct answer. Which of the following is not true about insulin? So there are four parameters are being given and we have to find out which one is incorrect as far as insulin is concerned. So let's see the structure of a insulin. Insulin, as we can see, is a peptide which is composed of two peptide chain A and B, which is linked together with a 2 disulfide bond. And there is one additional disulfide bond which is present in the A chain. It is released by the pancreas to lower the blood glucose level and it promotes the glycogen synthesis by activating the glycogen synthase enzyme. Once its job is done, it is being degraded by insulinase. So option A is incorrect, while option B, C and D are the correct about the insulin. Insulin is secreted by liver, muscles, gallbladder or pancreas. Insulin hormone that regulate the level of sugar in the blood is being produced by the beta cells of the isolate of Langerhans, which is present in the pancreas. So option D is the correct answer. Role of glucagon is to stimulate the formation of cyclic ATP in the liver cell, stimulate the formation of cyclic AMP in the liver cell, inhibit the formation of cyclic ATP in the liver cell, or inhibit the formation of cyclic AMP in the liver cell. Glucagon, it elevates the blood glucose, which is an opposite action of insulin, by activating a adenyl cyclase enzyme, and thus it increases the cyclic AMP production. So option B would be the correct answer, that glucagon generally stimulates the formation of cyclic AMP, as you can see in the figure, in the liver cell. So option B is the correct answer. Which hormone is also known as hyperglycemic hormone? That means which increases the glucose level in the blood. Glucagon, cholecystokinin, insulin or glycolase. Pancreatic glucagon, it promotes the glycogenolysis, that means conversion of glycogen to the glucose. It also promotes the gluconeogenesis, that means the production of glucose from a non-carbohydrate moieties. Eventually, both of these processes increases the blood glucose level. So glucagon is a hyperglycemic hormone, that it increases the glucose level in the blood. Which of the following is an example of anorexinic hormone? Endocannabinoids, cortisol, leptin or ghrelin. First of all, we should know what is anorexinic hormone. Any hormone which reduces the appetite or your feel for hunger, that is called as anorexinic hormone, like insulin, leptin, cholecystokinin, or even alpha melanocyte stimulating hormone. Ghrelin is the opposite of anorexinic hormone. So hence here the leptin would be the correct choice. Which is the main nitrogenous waste in humans? Urea, uric acid, ammonia, or ammonium nitrate. As we know, human is a ureotelic and main nitrogenous waste it releases is urea. So option A is the correct answer. The absorption of glucose in the digestive tract, that's very simple. Actually, the absorption of the glucose is being done in the small intestine by the absorptive cells through the secondary active transporters. So here the option A is the correct answer. The blood volume decreased when a mammal was bled rapidly. However, the cardiovascular changes resulting from hemorrhage could be maintained by the following compensatory mechanisms: increased cerebral flow, reduction of baroreceptor activity and stimulation of chemoreceptor, reabsorption of tissue fluid in the blood, or increased release of enkephalin and beta endorphin. So we have to find out the correct answer from the options given below. Basically what happens when a blood loss is high during hemorrhage, arterial pressure decreases and cardiac output is also decreases, which is resulted in a series of compensatory cardiovascular responses to restore normal functioning. If suppose ये blood loss जो हुआ है, if this is moderate, then basically the baroreceptor signaling takes place. But if there is a heavy blood loss, then baroreceptor signaling is diminished and chemoreceptor reflexes is being taken over. So B statement is correct. Now, decreased cardiac output led to the profound fall of blood pressure and heart rate. So the pressure in the capillaries also fall down and less amount of fluid is transduced from the tissue spaces. So reabsorption of tissue fluid in the blood takes place. So C statement is also correct. Now, due to the decreased cardiac output, the cerebral flow is being decreased rather than increased. So statement A is incorrect. Now, because of the diminution of the cerebral blood flow, we often observe fainting or unconsciousness. Now, as we know that the endorphin and enkephalin, these are the natural painkillers and mostly released by the brain to the blood. So definitely if the cerebral blood flow is reduced, then definitely their amount in the blood cannot get increased. So statement D is incorrect. Now, neurohormonal compensatory changes also involve the release of renin from the kidney and ultimately angiotensin II and aldosterone increases. Both of these hormones basically ensure the more retention of sodium and water from the urinary output, which further allow the reabsorption of tissue fluid in the blood. So only B and C statement is the correct, while rest of the statements are incorrect. In sickle cell hemoglobin, which one out of the following is true? A glutamate is substituted by a glutamine is substituted by a valine is substituted by a glycine is substituted by the valine. This is again one of the most simplest question which we are encountering since our lower classes. In sickle cell hemoglobin, a glutamate, which is a charged amino acid, is replaced by a hydrophobic long chain valine amino acids. So option A has to be the correct answer. Which of the following is not served by the plasma proteins? Blood clotting, oxygen transport, hormone binding and transport, or buffering capacity of the blood. Transport of oxygen is a function of hemoglobin, which is absent in plasma, rather present in RBC. So oxygen transport cannot be performed by the plasma protein, rather by RBC protein. The protein responsible for the formation of fibrin monomer is thrombin, prothrombin, heparin, or plasminogen. Thrombin, it converts fibrinogen to fibrin, which is an integral step in the clot formation. So option A is the correct answer, that thrombin is responsible for converting the fibrinogen to the fibrin monomer. During aerobic exercise, blood flow remains relatively constant within the brain, the heart, the skin, or the skeletal muscles. Aerobic exercise caused increased heart rate and cardiac output in most of the tissue except brain, which remain almost constant due to the autoregulatory mechanism. So option A is the correct answer. A single calmodulin protein can bind to how many calcium molecules? 4, 1, 3 or 2. Calmodulin is a small dumbbell-shaped protein which is composed of two globular domains connecting together by a flexible linker. It has two calcium binding sites in each of the protein lobe. Hence, in total, 4 calcium ions can bind in one functional calmodulin complex. So, option A is the correct answer. Calcitonin is secreted by parathyroid gland, pituitary gland, adrenal gland, or thyroid gland. Calcitonin is a hormone that is produced and released by the parafollicular cells of the thyroid gland. This hormone is basically responsible to regulate the calcium ion level and phosphate in the blood. So option D is the correct. The effect of IP3 activation is increase in intracellular calcium levels, increase in extracellular calcium levels, closure of calcium channels in ER, or activation of calmodulin. IP3 activate calcium ion channels on the cell membrane indirectly, which induces the release of calcium ions. Hence, IP3 causes a transient increase in intracellular calcium ion concentrations. Which of the following is not a peptide hormone? Glucagon, oxytocin, estrogen, and vasopressin. It's very simple. Estrogen is an example of steroidal sex hormone which is present in a female. So it is having an steroidal skeleton. So option C is the correct answer. Which of the following signaling molecule does not interact with cell surface receptor? Insulin, gastrin, glucagon, or testosterone? That's very easy. Insulin, gastrin, and glucagon are peptide hormones which actually generally interact with the cell surface receptor as they cannot cross the membrane. While on the other hand, testosterone, which is an steroidal hormone, do not bind to the surface protein and cross the plasma membrane to interact with the DNA. So here the option D has to be the correct answer. Select the true statement regarding nitric oxide, NO. NO is a vasoconstrictor. NO activates adenylate cyclase. NO activates guanylate cyclase, or NO stimulates platelet aggregation. Nitric oxide or NO is basically a vasodilator which inhibits platelet aggregation. So option A and D cannot be correct statement regarding NO. NO basically activates the guanylate cyclase activity which actually inhibits the adenylate cyclase activity. So option C is the correct answer. Prolactin is a peptide, steroid, nucleic acid, or vitamin. Prolactin is a peptide-based hormone produced in the pituitary gland. So, option A is the correct answer. Chemical messengers that are secreted by the cells into the extracellular fluid and affect neighboring target cells of a different types are typically known as autocrine, neurotransmitters, paracrines, or endocrines. Hormones are the chemical messenger secreted into the blood or extracellular fluid by one cell that affect the functioning of other cells. So option D is the correct answer, that chemical messengers that are secreted by the cells into the extracellular fluid and affect the neighboring target cells of a different cell types of cells are basically example of endocrines, which is commonly used for the hormone secreting cells. Propagation of the action potential through the heart is faster in the AV node, Purkinje fiber, SA node, or arterial muscles. Electrical activity that originated from the SA node is propagated via the His-Purkinje fiber network to the rest of the heart. It is the fastest conduction pathway present in the heart. So option B is the correct answer. What is the average resting potential of a mammalian cell? Plus 50 millivolt, plus 60 millivolt, minus 70 millivolt, or minus 50 millivolt? In most of the mammalian cells, the resting mammalian potential is around minus 0.05 volt or minus 50 millivolt. But this value may vary from minus 20 to minus 100 millivolt depending on the cell to cell. So normal average value of a resting potential of a mammalian cell would be minus 50 millivolt. Sphingomyelinase catalyze the conversion of sphingomyelin into phosphatidylinositol and ceramide, phosphatidylserine and ceramide, phosphocholine and ceramide, or acetylcholine and ceramide. Basically, sphingomyelinase catalyzes the hydrolysis of sphingomyelin to ceramide and phosphatidylcholine. So, option C is the correct answer. Tay-Sachs disease is caused by the deficiency of glucocerebrosidase, galactosidase, hexosaminidase, and sphingomyelinase. Tay-Sachs disease is a progressive neurodegenerative disorder which is caused due to the deficiency of hexosaminidase A, resulting in the accumulation of GM2 ganglioside. Just recall, we have just discussed the hexosaminidase A as a characteristic marker of lysosomes. So definitely Tay-Sachs disease is a lysosomal storage disorder. The disease, commonly called as lockjaw, is caused by Clostridium tetani, Mycobacterium leprae, Trypanosoma pallidum, or Bacillus pertussis. Another name of a tetanus is a lockjaw, which is caused by the Clostridium tetani, in which it is hard to open the mouth or swallow the food. So option A would be the correct answer. चलिए जी, हम लास्ट अपने फेज में आ गए हैं और अब तक अगर आपको हमारा कंटेंट पसंद आया है तो एक उम्मीद तो हम करते हैं कि आप सब्सक्राइब करेंगे एंड यू विल शेयर विद योर फ्रेंड्स, राइट? तो प्लांट बायोकेमिस्ट्री में आपके रूटीन क्वेश्चंस ही आएंगे। आपको प्लांट हार्मोन्स पे क्वेश्चंस आ सकते हैं, आपको प्लांट फिजियोलॉजी के जो क्वेश्चंस बनते हैं वो आ सकते हैं, तो आप प्लीज उनको गो थ्रू कर लीजिएगा। पास्ट ईयर के क्वेश्चन को जितना आप रेफर करेंगे उतना ही आपको आइडिया लगता जाता है कि किस तरह के क्वेश्चन बनते हैं एमएससी बायोकेमिस्ट्री के एग्जाम्स में। ओके, चलिए चलते हैं। Photon capturing moiety of chlorophyll molecule is known as porphyrin, magnesium porphyrin, chlorophyll B, or chlorophyll A. In chlorophyll, magnesium porphyrin is the moiety which functions to capture energy and store electron during photosynthesis. So, option B has to be the correct answer. The protein involved in photosynthesis is hemoglobin, collagen, rubisco, or albumin. Rubisco or ribulose biphosphate carboxylase is a universally present enzyme in all photosynthetic plants. This is very simple lower class question which you can remember. So option C has to be the correct answer. The precursor to IAA, or more often known as indole 3-acetic acid biosynthesis in plant is tyrosine, phenylalanine, tryptophan, or methionine. As you can see in the figure, the indole 3-acetic acid is the main oxygen produced by the plant using tryptophan as a precursor. So option C is the correct answer. A low oxygen to cytokine ratio led to, इसमें बड़ा एक इंटरेस्टिंग थियरी है। स्कूग एंड मिलर ने 1957 में ये बताया था कि हाई ऑक्सीजन और साइटोकाइन रेशियो जो होगा वो जनरली रूट रीजनरेशन को इंड्यूस करता है, जबकि लो रेशियो जो है सेम ऑक्सीजन एंड साइटोकाइन का लो रेशियो वो जनरली शूट को इंड्यूस करता है। रिमेंबर, हाई ऑक्सीजन साइटोकाइन रूट रीजनरेशन इंडक्शन, और अगर लो होगा तो शूट इंडक्शन। तो इसीलिए इसमें ऑप्शन जो होगा ए, दैट इज शूट फॉर्मेशन। The following condition decreases the level of denitrification. Denitrification जो है that are found when soil bacteria use nitrate for their respiration instead of oxygen, जो कि एक normal aerobic respiration का part। और ये जो प्रोसेस है ये एनहांस हो जाता है जब टेम्परेचर ज्यादा हो, ऑर्गेनिक मैटर ज्यादा हो, हाई पीएच हो, और इसीलिए इस क्वेश्चन में जो है डीनाइट्रिफिकेशन का जो लेवल है वो डिक्रीज कब होगा? हमने अभी जैसे बात किया हाई टेम्परेचर पे ज्यादा होता है, लॉगरेमिक मैटर की कंसंट्रेशन ज्यादा होगी तो भी होगा, हाई पीएच ज्यादा हो मतलब हाई पीएच होगा तो भी डीनाइट्रिफिकेशन ज्यादा होगा। तो लो कब होगा? लो पीएच पे। तो इसीलिए एट एसिडिक पीएच डीनाइट्रिफिकेशन का जो लेवल है वो लो हो जाता है। तो इसीलिए ऑप्शन डी हैज टू बी द करेक्ट आंसर। ओके, तो अब इस वीडियो को यहीं पे खत्म करते हैं। वी विश यू ऑल गुड लक फॉर योर एग्जाम्स। अगर आपको वीडियो पसंद आया तो प्लीज सब्सक्राइब आवर चैनल, हिट द बेल आइकन एंड कमेंट सेक्शन में भर भर के कमेंट्स कीजिए। प्लीज डू सब्सक्राइब अस एंड शेयर विद योर फ्रेंड्स। ओके, थैंक यू वेरी मच।